Hello friends, welcome to mega revision session of grassroots. Let us now talk about anatomy. Now in anatomy, very important is the plastid, and you should be knowing about all the kinds of the plastids that are there in the plant cell, like the leucoplast, etioplast, amyloplast, chloroplast, elioplast, proteinoplast, and the chloroplast. So plastid is quite important. You should be knowing about this cell organelle. Then how the proplastid develops into which kind of plastid. So that is important for the exam. Now let us talk about this next important topic, and that is secretory system that we see in the plants. So you should be now by knowing that the secretory system can be divided into external as well as internal. So external secretory system has got glandular trichomes, nectaries, hydatodes, and if we talk about internal secretory system, then it includes secretory cells, which are the individual cells. Then these individual cells can form cavities, and these cavities or ducts can be lysogenous and schistogenous. So what is lysogenous cavity? What is schistogenous cavity? How they are formed? Then about latticifers. So the latticifers, whether they are articulate and non-articulate. So these are the things that you should be thorough with. The other part that is important for your exam is the ergastic substances. In the ergastic substances, you should be knowing about the starch and how they are produced and where they are stored, the tannins, where they are stored, the proteins, as well as fat, oils, and waxes. Apart from it, you should also be knowing about the crystals, and the shape of the crystals is very important. And mostly the calcium oxalate crystal and calcium carbonate crystals are also important. Now, there are different shapes of the crystals like graphite, styloids, prisms, druses, crystal sands, cystolith. You should be knowing the genera in which they are present. So they might ask you about the plant species in which the crystals are found. Different crystals are found in different plants. So just have a look at them and prepare that. Now, the ergastic substances can be of three types, and you should be knowing what are the reserve substances, what are secretory substance, and what are excretory substance. That are important. Then you should be knowing about the cell types. Okay, types of cells, that is the parenchyma cells, collenchyma cells, and the sclerenchyma. Okay, so types of tissues you also have to know, and then the tetrachia the ideal form of parenchyma cells in which there are 14 faces and 8 in which 8 are hexagonal and 6 are quadrilateral faces. So this kind of form is rarely achieved, but this is the ideal form of the parenchymatous cell. Okay, so you should be knowing this. Then we move on to the dermal tissues in anatomy, and in dermal tissues you should be knowing about the epidermis, and the epidermis includes the cuticle. Then you should be knowing the epicuticular waxes and cuticularization, cutinization. This process you should be knowing. Apart from this, you should be also knowing the stomatas and the trichome. When we talk about the stomata, we also talk about the stomatal complex because we talk about the guard cells and the subsidiary cells. Then you should be knowing the types of the stomatal complex, like the anomocytic, anisocytic, parasitic, diacetic, and actinocytic. And here you can see the diagram also for these, and this is the graminaceous, that is monocot the monocot stomatal complex. So you should be knowing the basic differences between these stomatal complex, and that may fetch you a question in this part. And then we move on to the vascular tissues. Now vascular tissue, as you know, can be divided into primary vascular tissues and vascular tissues क्या होते हैं, xylem and phloem. So vascular tissues can be divided into primary and secondary. So you should be knowing both. And in the later diagram you can see this, this is the cut section of the trunk, and the rather root, it's a root, and the different sections of the root, what are these, and what is epiblem or the epidermis, how the root hairs are formed, and what is the steel that is present in the primary roots, and then what is cortex, and all these structures that you see in this diagram, you should be knowing about them, and how the endodermal cells are placed with suberin. Okay, so Casparian strips. You should be knowing these things because they can fetch you question over here. Then we move on to the individual components of xylem and phloem. Now you know that xylem includes tracheids, vessels, xylem parenchyma, and xylem fibers. So you should be knowing about them, as well as you should also know about the diagrams related to that, so that you know what are bordered pits, what are simple pits, how the vessels are formed through the tracheids, and what are tracheids, what are xylem fibers. Then we move on to the perforation plate part, or that is present in the vessel. So you should be knowing about the perforation plate. Then we move on to the second type of tissue, and that is called as a vascular tissue, that is the phloem. In the phloem, there the components are sieve tube elements, the sieve cells, and the companion cells, and then phloem fibers and phloem parenchyma. So you should have um, an outline of these components of xylem as well as phloem. Then in the sieve elements, we see that there are sieve plates that are present, and what are the different kinds of sieve plates. And you know that in details, that is in gymnosperms, the vessels are present. So this kind of the exceptions that are present, that you should be knowing. Okay. Then we talk about the different types of wood that we see in the case of when we talk about the secondary xylem. Okay, so the difference between the sapwood and the heartwood, the main difference is that in the sapwood there is no deposition of the secondary metabolites or the ergastic substances, whereas there is deposition of, in the heartwood there is deposition of tannin, resin, gummy substances, or you can say the ergastic substances in the central part of the stem. So that makes it heartwood. So the basic differences that you can see in the table, you can refer it this table and just clear your doubts, and this is important. And then we talk about the second type of wood, that is called the spring wood or the early wood, and the difference between the autumn wood or the late wood. Okay, so they might ask you about early wood and late wood, spring wood or autumn wood. So the both are synonymous that I have written over here, and these are the basic differences that you see, how the cambium activity differs in both, and what are the changes in the xylem and the phloem during the early spring season or the late autumn season. Okay, so those differences you can refer from this table, and they are important. Then we move on to the gymnospermic wood. In gymnospermic wood, we know that in cycas we see manoxylic wood, whereas in the pinus we see pycnoxylic wood. In wood, what is the state of the cambium, and in the monoxylic or the manoxylic, what is the state of the cambium, that you should be knowing, and which one is more useful and which one is less useful. You can refer this table, and this might fetch you a question in exam, because this is a portion that is also present in the gymnosperm, and here it is also present. So this can fetch you a question. Then comes the part where the wood is under stress. When the plant is under stress, so two kinds of woods are seen, and these are the tension board and the compression board. So, what are these, and what is the difference occurs when an angiospermic plant is under stress, and what is the difference occurs when a gymnospermic plant is under stress? So, compression board and tension board has got some differences, whether they are present in a angiospermic plant or whether they are present in a gymnospermic plant. So, those differences you should know कि किसमें जिम्नोस्पर्मिक में ज्यादा होता है, या एंजियोस्पर्मिक वुड में टेंशन वुड ज्यादा बनती है, या कंप्रेशन वुड. That you see, and just have a look at the bottom part also, where I have included about the tension wood and the compression wood. Let us now talk about the primary growth. And before moving on to the exact secondary growth, you should be knowing about what are the parts of the root tip, and how the root tip brings about the primary growth in the how it extends the root towards the deeper depth of the soil. Okay, so there are different regions that you should be knowing about. And then we talk about the shoot apex. What are the different regions of the shoot that is important? Okay. Then tunica carpus theory is also important. Okay, so just have a look at the tunica corpus theory. This will also fetch you marks. Then types of cambium is also important, where the apical meristem is formed or where it is present and what is the use of the apical meristem. Then we talk about intercalary meristem. What is the use of this intercalary meristem? What is lateral meristem and what is the use of lateral meristem? So type of meristem and type of cambium you should be knowing about. Then what is the difference between the primary growth that takes place and the secondary growth? What are the changes that occurs when a plant undergoes the secondary growth from the primary growth? Okay, so how the periderm is formed, how the cork cambium emerges. So you should be knowing these differences. That is important. And then we move on to the next phase of our analysis, and that is the secondary growth. In the secondary growth, how the interfascicular and the fascicular cambium joins together to form a complete cambium ring, and then this cambium ring becomes active and cuts out xylem towards inside and phloem towards the outside. That you should be knowing, and that is called the secondary growth, and that will be important with the exam perspective. Okay, so just have a look at that, and that will be useful for you. Then we move on to the next part, and that is abnormal secondary growth. So what I've done is I've classified the abnormal secondary growth into how, on what basis the abnormal secondary growth takes place. Like we talk about the primary growth. This primary growth takes place in the apical meristem or the root apex. Okay, the primary growth takes place, and this leads to the increase in the height of the plant or increase in the length of the root. Okay, then comes the secondary growth. Now here the meristem involved is the intercalary meristem. It increases the girth of the plant, so the thickness of the plant is increased. This is the outcome of secondary growth. Now we talk about anomalous or the abnormal secondary growth. This can be because of the adaptive nature or the non-adaptive reasons. And the adaptive is to cope up with the external environment. That means there is some stress because of which the abnormal or the anomalous secondary growth is taking place. Now there can be several other reasons, like there is no adaptive force that is acting on the plant, or we can say that the plant is not under stress. Even that also, then also it is showing the anomalous or the abnormal secondary growth. So in that case, there is variation in the cambial activity which brings about this anomalous secondary growth. Then what I've done is I have categorized in different in ways in which these anomalous secondary growth takes place. Okay, so abnormal functioning of the cambium, and this basically talks about the non-adaptive reason. This table talks about it. Okay, so the abnormal or the anomalous secondary growth takes place because of the abnormal functioning of the cambium. Now, this kind of anomalous secondary growth you will find in Tinospora stem, Bignonia stem, Bauhinia stem, and Doxantha stem. So, if a question arises, then you should be knowing that in Doxantha stem, the anomalous secondary growth takes place, and because why it takes place, because of the, and also the reason for all these four, the reason is this. Okay, so the reason, they might ask you the reason, or they might ask you the otherwise. Okay, because of the abnormal functioning, in which of the plants you will see the anomalous secondary growth, or they might ask you out of these four plant, or they might ask you in Bauhinia, out of which reason this anomalous secondary growth takes place. So you should be knowing that because of the abnormal functioning of the cambium. Then this is the first reason. Now let us talk about the second reason, and the second reason is unusual position of the cambium. The cambium is unusually placed. Now anomalous secondary growth takes place in Thaunoia stem and Sergenia stem. So these are the two plants that show the anomalous secondary growth because of the unusual position of the cambium. Then when we talk about the third reason why this anomalous secondary growth takes place in Boravia stem. So anomalous secondary growth takes place in Boravia stem because of the formation of more than one cambial rings. So there are more than one cambial ring that are formed, because of which what happens, anomalous secondary growth takes place. Then anomalous secondary growth takes place in Salvadora stem also, and this takes place because of the interxylary phloem. So this is the fourth reason why the anomalous secondary growth is seen in Salvadora. The last fifth reason because of which this anomalous secondary growth is seen in Dracaena. Okay, so why? Because there is secondary growth in the because Dracaena is a monocot, so we talk about the secondary growth in the monocot stem. So this is a separate thing, but I have put in here so that it is easier for you to categorize.
Hello, everyone. Let us now talk about plant physiology.
Now, uh, essential elements. What are these? What are essential elements? And what is the criteria on which the elements are classified as essential elements? Now, to tell you, there are total 17 essential elements. Out of these 17 essential elements, 3 are non-essential elements and the 14 are mineral elements. Now, the example are given here like carbon, hydrogen, oxygen are non-mineral elements, whereas the other one are the mineral elements like that include zinc, potassium, magnesium, boron, chlorine. Okay, so these are 14. And non-mineral elements like carbon, hydrogen, oxygen, these are 3. So total there are 17 essential elements. You should be knowing what is the criteria that divides the elements into essential elements. Okay, then what is the source that I have already mentioned? You can see here. Then we talk about the essential mineral element or the mineral nutrients. And this concept was given by Arnott and Stout in 1939. So this is also important. Then we talk about the mineral elements. The mineral elements that are 14. So we are talking about this factor. So these can be divided into two main categories. One is macro element, the other one is micro elements. Macro elements are total 6. That is nitrogen, potassium, phosphorus, calcium, magnesium, and sulfur. Okay. Then the other one are, you can see in your slide that these are 8. So just have a look at them and what is the requirement in which they are needed. You must have heard about NPK fertilizer. That is that is greatly used in the agricultural field. So this comes under the all these three element comes under macro elements. So these are macro elements. That's why they are required in large amount. Then other these 6 macro elements are divided into what? They are also divided into two categories. The one is the primary nutrients, which includes the nitrogen, potassium, and phosphorus. Okay, these three is NPK. Now the other one is the calcium, magnesium, and the sulfur. These are called as secondary nutrients. Okay, so the again forms the group that you should be knowing about. Let us now talk about the macronutrients. Before talking about the macronutrients, let me first tell you about Lebesgue law of minimum. So this is a very important law that you should be knowing about. You can expect a question from here. What he said, first of all, this was developed by Justus von Liebig, and he said that the growth of the plant is dependent upon the element or the nutrient which is supplied in least quantity, and this should be in relation to the need of the plant. Okay, so there can be several elements that are present in the nutrient medium, but the element that is present in the least quantity in relation to the need of the plant, that will be the element that is controlling the growth of the plant. Unless and until you increase the amount of that element, the growth of the plant will not resume. So you should be knowing about the experiment that was done on the plant to prove this law, and this will be important for your exam. Then let us talk about the macronutrients and the micronutrients. The macronutrients are three. I've told you that one is the nitrogen, the second one is phosphorus, and the third one is potassium. So nitrogen, what is the function of nitrogen inside the plant, and what are the deficiency symptoms that will appear on the plant if the plant is supplied with the less of the macronutrients. So you can have the look at the table, and you can also see what are the deficiency syndromes and what is the function of that particular macronutrient. So these are macronutrients. These are called NPK together. They also are part of what? Fertilizer. And the farmers use this fertilizer to in the field. Then we talk about the micronutrients. These are also three: calcium, magnesium, and the sulfur. And in this table also you will find the various functions that are controlled by these macronutrients. And these macronutrients or the secondary nutrients. So what we have done is we have divided the macronutrients into two parts: primary nutrients and secondary nutrients. Primary nutrients में NPK आ गया और secondary nutrients में calcium, magnesium and sulfur आ गया. Then you should be knowing about the functions, individual functions of these in the plant. and what are the deficiency syndromes or the deficiency symptoms that will appear on the different parts of the plant if the plant is supplied in less quantity with calcium, magnesium, and sulfur as per the requirement of the plant. Okay, so that just you just go through this table, and that would be helpful for you to attempt the question in exam. Now let us move on to the micronutrients. So these micronutrients are three. So six were the macronutrients, and three out of those six macronutrients, three were the primary nutrients and three were the secondary nutrients. Now we are talking about three micronutrients. These are molybdenum, manganese, and zinc. So these are less required as compared to the macronutrients. And in macronutrients, primary nutrients are more required as compared to the secondary nutrients. So total six macronutrients and three micronutrients. So micronutrients, just go through the functions and the deficiency symptoms. So the next phase of the three micronutrients are the boron, iron, and copper. So these are the three mm, micronutrients that you need to know, and these are the functions that they control inside the plant, and these are the deficiency symptoms. So just go through this table and just see the function so that you may attempt the question in exam. Now let us talk about the mechanism of absorption. Now you must have heard about the active and the passive transport. So what are these and which one utilizes the ATP? So you should be knowing about it. Then we talk about the transport proteins, and these transport proteins can be selective channels. And then what is selective channels? What are carrier proteins, and what are the specific carrier proteins that you see in the plant? So you should be knowing about all this. Then we talk about passive transport. What is passive transport? You know that in which there is no utilization of ATP. And active transport may we utilize ATP, or the plant utilize ATP to transport materials from one side to the other. Okay, so this happens always against the concentration gradient, whereas passive transport occurs down the concentration gradient. So these are the important topics that you need to revisit or you should know. Then we talk about the movement of water inside the plant, how the roots absorb the water, and when the water enters into the plant, the water either moves through symplastic pathway or it moves through apoplastic pathway. So the apoplastic pathway is shown in the diagram in the white color, whereas the symplastic pathway is shown in the diagram in the gray or the black color. Then you'll observe that after endodermis, inside the endodermis there are Casparian strips which are laden with suberin. So that you have studied in anatomy also. Now from there, the water is forced to move inside the cell wall, and then water moves through the symplastic pathway, and then it enters the xylem over here. So this is how the movement of water is inside. Movement of water takes place from the root hair, from outside to the root hair, inside the root hair to the xylem. So after xylem, it is pulled through the vessels and tracheids up to the leaf, towards the leaves, because of the transpirational pull that you already know. Then we talk about the transport in the vascular plants. Now the vascular in the vascular plant, you know that there are vascular tissues. Now two kinds of vascular tissues, that is xylem and phloem. Now you already know that xylem transports the water, whereas phloem transports the food. Now, you should know about the bulk flow. What is bulk flow and what is diffusion? You should also know the difference between the two. What is bulk flow and how it regulates the transpiration and translocation. So that is required for you to know. Then we talk about ascent of swab. So what is the meaning of ascent of swab? That is the upward movement of minerals and water against the concentrational, against the gravitational force. And this occurs from the root to the apical part of the plant. So this is called as ascent of sap, and the mechanism of ascent of sap. The mechanism is also important. Mechanism of ascent of sap is also important, and that mechanism is called as cohesion tension model, which is also called as transpirational pull, which was proposed by Dixon and Joly. Okay, you should be knowing this. This is very important for the exam. And then let us now talk about the materials that are translocated in phloem. So translocated solute is what? This is the carbohydrate, and sucrose is the most commonly translocated sugar. This is very important. You can expect a question from this slide. So just pay attention. Now the carbohydrates are translocated as non-reducing sugars. What is then what the question arises, what is phloem sap? Then phloem sap includes the organic molecules, which includes the amino acids, the plant hormones, and the inorganic ions. Okay, so organic molecules are the major part. The rest of the things are also present in the phloem sap. So let us now talk about the water potential. So what, first of all, what is loading? So loading is the transport of sugar into the sieve element. What is unloading? Transport of sugar out of the sieve element. So you should be clear with what is unloading, what is loading. Then we talk about the water potential, and you know that water potential is the summation of water solute potential, pressure potential, gravitational potential, and matrix potential. So this solute potential is given by a formula which is called as Van 't Hoff equation, and you should be knowing the Van 't Hoff equation, that is minus CIRT. Now C stands for the molar concentration of solute, I stands for ionization constant of the solute, whereas T is the absolute temperature in Kelvin, and R is the gas constant. So you should be knowing these terms, and this is important. Finally the equation becomes the water potential is equal to the solute potential, and the pressure potential is also called as Turgor potential. Okay, so Turgor pressure. So the pressure is the Turgor pressure. The pressure that is applied on the walls of the cell wall. Okay, so basically the pressure that is felt on the cell wall. So since the matrix potential and gravitational potential values are very negligible, so we avoid them. So the equation finally becomes water potential is equal to the solute potential plus the pressure potential. Now let us just have a look at the, you should also pay attention to the mechanism of loading and unloading, how the active loading occurs, and how the active unloading occurs at sink and source. Okay, so how the sucrose moves from the source, and how it falls, or it is unloaded at the sink region. So you can have a look at this diagram. In this diagram, they have beautifully, what I've done is I've put in two diagrams. So if by looking at these two diagrams and the details that are mentioned in the diagram, if you read them, then you will be clear with the idea of loading and unloading. Don't make any mistake in this. And then you can just see simultaneously how xylem vessels are present, and the water movement occurs from the vessel to the sieve tubes of the phloem. So, and in the sieve tubes, the loading and unloading process is going on. So translocation of transpiration of water and translocation of solute is shown simultaneously in this figure. So they, have a look at this figure also. And then we talk about the photoperiodism, in which we talk about short day plants and long day plants. You know that short day plants flower when a period of darkness, or the period is uninterrupted and it exceeds a critical day length. So you should be knowing what is critical day length, what are short day plants, and what are long day plants. So what are long day plants? Long day plants are those plants which flower when a period of dark is less than the critical day length of the sunlight. Okay, so you should be knowing the examples also. Just have a look at this diagram. This diagram explains beautifully the concept of photoperiodism. Okay, okay, guys. Best of luck for your exam, and whatever topics I have discussed with you, kindly go through them, and the details that are mentioned in the slides, and just revisit your notes, and then clarify your doubts. Okay, good luck and best of luck.
नाइट्रोजन फिक्सेशन जो है ना सिर्फ माइक्रोबायोलॉजी में बट बॉटनी में भी बहुत क्वेश्चन इनवाइट करता है, वन ऑफ द मोस्ट इंपॉर्टेंट टॉपिक। और नाइट्रोजन फिक्सेशन आसान टॉपिक नहीं है, इसमें बहुत रैंडम क्वेश्चंस बनते हैं, ठीक है ना। तो इसमें बहुत सारी चीजें पढ़ने के लिए होती है। नाइट्रोजन फिक्सेशन होता क्या है? नाइट्रोजन फिक्सेशन एक प्रोसेस होता है जिसमें हम जो नाइट्रोजन है, जो कि एटमॉस्फेरिक नाइट्रोजन है, उसको यूज करते हैं, एंड देन हम उसको इस फॉर्मेट में कन्वर्ट करते हैं बाय यूजिंग नाइट्रोजन फिक्सिंग बैक्टीरिया, सो दैट इट कैन बी आइदर कन्वर्टेड टू अमोनिया और सम अदर फॉर्म्स जिनको हम बाद में यूज कर सकें। अब दिस अमोनिया को हम विद द हेल्प ऑफ दिस प्रोटॉन्स हम इसको कन्वर्ट कर देते हैं अमोनियम में। अब ये जो अमोनिया है, इसको नाइट्रीफाइंग बैक्टीरिया या तो नाइट्रेट में कन्वर्ट कर सकते हैं, और या फिर इस अमोनिया को हमारे प्लांट में भी ड्राइव कर दिया जाता है, जहां पे हम इसको यूज करेंगे for the different purposes, for amino acid making, for nucleotide making and any other nitrogen containing compound. ये नाइट्रेट को डीनाइट्रीफाइंग बैक्टीरिया वापस से रिटर्न कर सकते हैं और उस प्रोसेस को हम डीनाइट्रिफिकेशन करते हैं. लेट्स हैव अ लुक ओवरऑल नाइट्रोजन साइकिल, जो कि इसका एक सिग्नेचर है. इसकी जो सिग्नेचर है वो स्टार्ट होता है नाइट्रोजन से, जैसे अभी हमने बात करी एटमॉस्फेरिक नाइट्रोजन से. और नाउ इन ऑक्सीडेटिव एनवायरनमेंट या एनारोबिक एनवायरनमेंट में किस में हो रहा है उस पर डिपेंड करता है ये नाइट्रोजन साइकिल कैसे चलेगी. अगर हमारा ऑक्सीडेटिव एनवायरनमेंट है जिसमें कि हमने अमोनिया बना लिया, यानी अपर लेयर ऑफ द सॉइल्स, यानी कि टॉप सॉइल से ऊपर, तो देन इस अमोनिया को अगर हम नाइट्रेट में कन्वर्ट करते हैं तो नाइट्राइफिकेशन कहते हैं, जो कि बाद में नाइट्रेट बन सकता है. और इस नाइट्रेट को भी हम यूज कर सकते हैं एज ए नाइट्राइट. अब नाउ ये जो नाइट्राइट है जो कि सॉइल में अब पहुंच चुका है, इसको यूज करने के, इसको वापस से डीनाइट्रिफिकेशन करने के या फिर इसको वापस से नाइट्रोजन में कन्वर्ट करने के कई सारे मेथड हो सकते हैं इन द एब्सेंस ऑफ ऑक्सीजन. या तो ये डायरेक्टली कन्वर्ट कर दिया जाए बाय डीनाइट्रीफाइंग बैक्टीरिया, या फिर एक और तरीका है, वो क्या है कि हम इसको फर्स्ट कन्वर्ट करें नाइट्रस ऑक्साइड या फिर N2O में, और देन इट इज बीइंग कन्वर्टेड इनटू N2. इस प्रोसेस को हम बोलते हैं एनामॉक्स. एंड वापस से ये N2 जो है वो रिलीज कर दी जाती है, या तो फिर एनवायरमेंट में जा सकती है या फिर ये अमोनिया के फॉर्मेट में फिर से फिक्स कर दी जाएगी. अगर हम इस अमाइनो ग्रुप को या NH3 ग्रुप को अगर हम फिक्स कर देते हैं प्रोटीन्स में, तो उस प्रोसेस को हम बोलते हैं अमोनिफिकेशंस. जबकि अगर हमारा नाइट्राइट फिक्स हो रहा है प्रोटीन्स में, अमाइनो एसिड्स में, तो उसे बोलेंगे हम एसिमिलेशंस. तो ये जो टर्मिनोलॉजीज हैं इनको आप गो थ्रू कर लीजिएगा एक बार अच्छे से. नाइट्रिफिकेशन क्या है, डीनाइट्रिफिकेशन क्या है, नाइट्रोजन फिक्सेशन किसको बोलेंगे, अमोनिफिकेशन किसको बोलेंगे, एसिमिलेशन किसको बोलेंगे और एनामॉक्स किसको बोलेंगे, ये स्ट्रेट अवे क्वेश्चंस हैं. और इसमें जो ऑर्गेनिज्म जो रोल प्ले करते हैं वो बैक्टीरियाज हैं. दे हैव द कैपेसिटी टू फिक्स दिस नाइट्रोजन. रिमेंबर, दिस नाइट्रोजन जो है ये एक ट्रिपल बॉन्ड सिस्टम है, और इसको अगर हमें रिड्यूस करना है तो हमें बहुत एफर्ट डालने पड़ते हैं. इसकी जो एक्टिवेशन एनर्जी है वो बहुत हाई होती है. यू रिक्वायर अ लॉट ऑफ एटीपी, लॉट ऑफ अदर सिस्टम इन ऑर्डर टू रिड्यूस इट. और स्टेप्स क्या होते हैं, पहले नाइट्रोजन जो मॉलिक्यूल है वो डाई इमाइन बनता है, फिर हाइड्रोजन बनता है और बाद में 2 मॉलिक्यूल ऑफ अमोनिया बन जाता है. ये जो नाइट्रोजन फिक्सिंग है ये स्पेशल बैक्टीरिया करते हैं, स्पेशल माइक्रोब्स करते हैं, और उनको हम नाइट्रोजन फिक्सिंग बैक्टीरिया भी बोलते हैं. और यहीं पे क्वेश्चंस आते हैं कि कौन कौन से नाइट्रोजन फिक्सर्स हैं. या तो वो बैक्टीरिया हो सकते हैं, फॉर एग्जांपल राइजोबियम, फ्रैंकिया, एजिटोबैक्टर, माइकोबैक्टीरियम, एजोस्पाइरलम, बैसिलस. या फिर वो आर्किया हो सकते हैं, मिथेनोकोकस, मिथेनोमाइक्रोबाइल्स, राइट. या फिर वो साइनोबैक्टीरिया होते हैं जिसपे क्वेश्चंस हमेशा एनाबिना या नॉस्टॉक पे मोर ऑफन पूछे जाते हैं. ये जो नाइट्रोजन फिक्सर्स हैं ये या तो फ्री लिविंग हो सकते हैं या फिर एक एसोसिएटिव कंडीशन में हो सकते हैं. या तो वो सिम्बायोटिक रिलेशनशिप में रह सकते हैं या फिर वो एनारोबिक फ्री लिविंग में रह सकते हैं, और उनके एग्जांपल हमने यहाँ पे मेंशन किए हैं. तो स्ट्रेट अवे क्वेश्चंस यहाँ पर यही पूछे जाते हैं. फ्री लिविंग एरोबिक नाइट्रोजन फिक्सर्स के एग्जांपल, फ्री लिविंग एसोसिएटिव नाइट्रोजन फिक्सर्स, सिम्बायोटिक रिलेशनशिप में कौन रहता है, लेग्यूम्स में रहता है या एल्डन ट्रीज पे रहते हैं, फ्री लिविंग एनारोबिक नाइट्रोजन फिक्सर्स कौन से हैं, एग्जांपल्स. ये क्वेश्चंस आएंगे और इनके आप देखिएगा CUET PG लास्ट ईयर में भी बहुत क्वेश्चन आए हैं. इस टेबल को आप यूज कर लीजिएगा. इसके बाद हमने वन लाइनर्स यहाँ पे मेंशन किए हैं. इस पे भी अक्सर क्वेश्चंस आ जाते हैं इन वन लाइनर्स में. वन लाइनर बड़े इंपॉर्टेंट होते हैं. आपको पूरा सारा टॉपिक पढ़ने की जरूरत नहीं है, अगर ये एक लाइन पढ़ ली आपने तो भी आपको क्वेश्चन बन जाएगा. और अल्टीमेटली जब आपका एंट्रेंस हो रहा होता है तो वन लाइनर्स आर द ब्लेसिंग्स. पूरा बुक ना पढ़ने की बजाय आप एक इंपॉर्टेंट वन लाइनर्स पढ़ लीजिए. इस तरह के वन लाइनर्स को हमने गैदर किया है एक इंपॉर्टेंट बुक से, जिसको कि हम लिटरेचर को रिव्यू करेंगे. ठीक है, हम बताएंगे भी आपको कहां से किया है. राइस प्रोड्यूसर जो है जनरली वो एक्वेटिक एजोला फर्न्स को ऐड कर देते हैं एज अ ग्रीन मैन्योर, और जो कि क्या करता है, इट प्रोवाइड्स अ हैबिटेट फॉर एनाबीना, और जो कि एनाबीना नाइट्रोजन फिक्सिंग प्रॉपर्टी शो करता है. एनाबीना और नॉस्टॉक दोनों साइनोबैक्टीरिया हैं, ब्लू ग्रीन एल्गी हैं, और फ्री लिविंग नाइट्रोजन फिक्सर्स भी है. जो फ्री लिविंग नाइट्रोजन फिक्सिंग एरोबिक बैक्टीरिया हैं उनके एग्जांपल हैं आपके एजिटोबैक्टर, बिजर्निका एंड क्लॉस्ट्रीडियम. रोडोस्पाइरलम पे भी क्वेश्चन आए हुए हैं, विच इज एन एनारोबिक एंड बैसिलस एंड विच इज फ्री लिविंग. राइजोबियम नाइट्रोजन फिक्सर्स है रूट नोड्यूल्स में, जबकि एजोराइजोबियम जो है वो स्टेम नोड्यूल्स का नाइट्रोजन फिक्सर्स है. तो ये क्वेश्चंस बनेंगे, आप देखिएगा, नोट डाउन कर लीजिएगा, हेल्प ही होगी आपकी। अब जो नाइट्रोजन फिक्सिंग का जो अप्लाइड वर्जन है उसको हम बोलते हैं नाइट्रोजन फिक्सिंग बायोफर्टिलाइजर्स। बायोफर्टिलाइजर्स या फर्टिलाइजर्स होते क्या है? सॉइल्स में आप क्या ऐड करते हो? एनपीके ना। तो एन मतलब नाइट्रोजन, पी मतलब फॉस्फेट और के मतलब आपका पोटेशियम। इसमें से यानी कि नाइट्रोजन आपको ऐड करना है, यूरिया आप ऐड करते हो या फिर कुछ और मॉलिक्यूल। अगर हम ये जो बैक्टीरिया हैं इनको ऐड करें विच विल फिक्स द नाइट्रोजन, तो वो बन जाएंगे हमारे बायोफर्टिलाइजर। जैसे फॉर एग्जांपल एनाबिना, अभी हमने बात करी थी, एनाबिना इज वन ऑफ द ग्रेट बायोफर्टिलाइजर। और यही स्ट्रेट अवे क्वेश्चन भी आता है कि बायोफर्टिलाइजर्स होते क्या है, उसके सबसे बेस्ट एग्जांपल क्या है। एट टाइम्स ये जो बैक्टीरिया हैं ये हमारे को टू फोल्ड बेनिफिट्स देते हैं। ना केवल दे आर बायोफर्टिलाइजर, दे आर प्रोवाइडिंग अस न्यूट्रिशन, बट ऑल्सो कभी कभी ये ऐसे टॉक्सिन्स भी रिलीज करते हैं जो कि ऐसे पैथोजेंस या ऐसे पैरासाइट्स जो कि प्लांट्स के लिए डेंजरस हो सकते हैं, निमेटोड्स या कुछ और ऑर्गेनिज्म, वो उनको सर्वाइव नहीं करने देते, उनको मार देते हैं। तो यूजिंग बायोफर्टिलाइजर इज अ टू फोल्ड बेनिफिट्स, ठीक है। तो एनाबिना अक्सर बायोफर्टिलाइजर्स के नाम पे क्वेश्चन बन के आता है। फाइटो हार्मोन्स जो होते हैं वो प्लांट बेस्ड हार्मोन होते हैं जो प्लांट की ग्रोथ के लिए बड़े इंपॉर्टेंट होते हैं। और यहां पर हमने आपको टेबल दी है, याद करिएगा फाइटो हार्मोन्स पे क्वेश्चन आपको बॉटनी में भी मिलने वाले हैं और जूलॉजी में भी मिलेंगे। और आपको 5 से 6 फाइटो हार्मोन्स, उनके केमिकल नाम, उनके फंक्शंस और कहां वो बनते हैं, कहां पे एक्ट करते हैं, उस पे क्वेश्चंस आएंगे। आप केमिकल के लिए आप ये वाली टेबल यूज कर सकते हैं, जबकि मेन फंक्शंस के लिए अगर हम डिस्कस करें तो ऑक्सिन का क्या फंक्शन होता है? ऑक्सिन का फंक्शन होता है इट स्टिमुलेट्स द स्टेम इलोंगेशन एट लो कॉन्सेंट्रेशन। और भी फंक्शंस है, आप पढ़ लीजिएगा। साइटोकाइनिन जो है उनका क्या इंपॉर्टेंट है? दे अफेक्ट रूट ग्रोथ एंड डिफरेंशिएशन, सेल डिवीजन को स्टिमुलेट करते हैं एंड दे आल्सो स्टिमुलेट द जर्मिनेशन, सेनेसेंस को दो वो डिले करते हैं। ये क्वेश्चंस बनेंगे स्ट्रेट अवे। जिब्रेलिन जो है वो फ्लावरिंग को डेवलपमेंट जो है उसको एनहांस करता है और फ्रूट डेवलपमेंट को भी स्टिमुलेट करता है। जबकि जो एब्सिसिक एसिड है वो आपका इनिबिशन करता है ग्रोथ का, स्टोमेटल की जो ओपनिंग एंड क्लोजिंग है उसको अफेक्ट करता है और सीड डोरमेंसी को प्रमोट करता है। इथिलीन जैसा कि हम सब जानते हैं वो फ्रूट राइपनिंग के लिए जरूरी है और ऑक्सिन का जस्ट अपोजिट एक्शन करती है। ये जो टेबल है ये वन पेज टेबल, ये आपको कुछ क्वेश्चन जरूर देकर जाएगी, जरूर देकर जाएगी बॉटनी एग्जाम्स में भी और आपके माइक्रोबायोलॉजी एग्जाम्स में भी। इसको नोट डाउन कर लीजिएगा और एक बार एग्जाम देने से पहले इसको जरूर देखिएगा, ठीक है। चलो,
Think about taxonomy. Now, in taxonomy, you should be knowing about the types of classification that are important, like the artificial, which includes the linear one, the natural system of classification, like the Bentham and Hooker and all, then phonetic classification, which recognizes the overall similarity from different, the data comes from different fields. Then phylogenetic, that is based on evolutionary lineage or the evolutionary classification per se that later developed. Okay, so you should be knowing about all the types of classification that is important and that can fetch you a question over here. Then the second important part is the developmental phase or the levels of the systematic. The first phase is the exploratory or the pioneer phase. The second phase is your consolidation phase or the systematic phase. The third one is the experimental or the biosystematic phase. And the fourth is the encyclopedic or the holotaxonomic phase. You should be knowing in which of the areas of the world or which regions of the world are experiencing which of these phases, that you should be knowing. Okay, and what happens, which kind of keys or floras or monographs are prepared in this in a particular phase, that you should be knowing. Then there are few key important topics that I want to tell you about, and that is you should be knowing the definition of biosystematics. What is biosystematics? You should also be knowing about chemotaxonomy. What is chemotaxonomy? How this chemotaxonomy is used in solving the queries of taxonomy, or in correcting our mistakes while when we have done the classification. So through chemotaxonomy we can change the classification. And so that is important. You should be knowing the definition. Then phylosystematics or phylogenetics. Then numerical taxonomy. What is taximetrics and cladistics techniques? That you should be knowing. That is the basic thing that that may ask you in tomorrow's exam. Then you should be knowing about the latest taxonomical tools, or the computer aided tools that are present in the today's world, and that is index nominum genericorum, that you should be knowing what is it. Then word biodiversity database, this is database. Then who developed it, who maintains it, what it is, and to which museum it is related, you should be knowing that. Then you should be knowing, you should be having a rough idea about international plant name index, what it is and what it contains, how many names it contains, that you should be knowing. Then what is tropics, this is very important, who maintains it and what it is, what is the significance of tropics, that is important. And that then what is electronic list and the news group, that is also very important. Just try to have these things clear in your mind, you because these things can fetch you questions in tomorrow's exam. Then you should be knowing about the hierarchical system of classification. This is that the that each level represents a taxon. Then you should be knowing what is a taxon, then what is a taxonomic group, and what is the difference between a taxonomic group and a taxon. Then this is the hierarchical system of classification, which one is which out of like suppose if I ask you a question, then kingdom, in the kingdom of this taxon kingdom, which is the highest kingdom, this is the most dissimilar, or this is the most similar. So this is the most dissimilar, or you can say this is the most inclusive group, whereas a species is the most similar group, but it is the most least inclusive group. So it has got individuals which are looking like to each other. Okay, morphologically similar organisms will be placed, or populations are placed in the species. So then we talk about the taxonomical nodes, aids that are important. We talk about herbarium. What is the size of herbarium? Where are the herbariums are kept? What is the meaning of herbarium? That is herbarium specimen that you mostly think about. Herbarium is a place. Okay, that differences you should be knowing about. Then what is botanical garden? Why they are maintained? Then what is a manual that is being prepared? In which phase of the taxonomic development, or the pioneer phase में होता है या दूसरे phase में होता है, that you should be knowing. Then what is flora, what is monograph, ये सब आपको पता होना चाहिए. Then this is for zoology part, you can leave this. But what are taxonomic keys, bio polyclave, multiclave, ये सब keys के बारे में भी थोड़ा सा आपको, you should be having some information about it. Then we talk about the type method. In type method, we talk about different the taxonomic evidence that a author submits while proposing a name, or while proposing any change. So a author submits a herbarium specimen or a taxonomic evidence, that is called type. Okay, so there are different types. What is a holotype, what is isotype, lectotype, syntype, neotype, एक paratype भी होता है. But these are the main ones, so you can see them and just have a look at this and try to clear in your mind, because this can also fetch you a question in the last exam. I have seen the question from here, from this slide. And then let us talk about the different families, the angiospermic families that are important. And I have discussed few families, you can just have a look at them. And from here this can fetch you a question. And the important family, the first important family that I'm going to discuss is Ranunculaceae, and this is commonly called as buttercup family. And in this, the stamens are numerous and they are spirally arranged, and the foliar spur is characteristic feature which functions as nectary. This is very important, and these are the important points I have highlighted with a bold. Okay, so but for you, you should be knowing, you should have just go through this and try to remember the genera that are present in them. Like Ranunculus is a type genus of Ranunculaceae. Then Nigella, Caltha. Then you should be knowing about the names of the different specimens that are present in this family. So let us now discuss the second family, and that is Caryophyllaceae. The common name of this family is carnation family. Okay, then the important part is that the outer wall of the flower has got 4 to 5 stamens, and they are modified as petals, now which are bilobe differentiated as basal claw and apical blade. So these are the question from these kind of details, they might ask you a question. Then the representative are dianthus, silene, agrostema, Okay, so these are the representatives of this family. Now, the androgynophore is very important, and this is the key aspect of this family, and you should be knowing about it. And they might ask you, like, they might put up a question like this, that in which of these they will give you four options. Now, in these four options, they will give you four different families, and they might put a question like, uh, in which of these family you will find androgynophore. Then you should be knowing that Caryophyllaceae has got androgynophore. So this kind of question can be put up. So this was the second family that we wanted to discuss, and the third one is the Leguminosae family or the Fabaceae family. In this, the you should be knowing about how the role of it in the nitrogen fixation, and then what are the different um, members of this family. And before discussing the members, what are important is the three subfamilies are very important, and this is Caesalpinoideae, Mimosaidae, and Papilionoideae. Okay, so these are the three you can say subfamilies which are placed under Fabaceae or the Leguminosae. So let us now talk about these families. And these the first one to discuss is the Mimosaidae. And in Mimosaidae, you can see that the leaves are bipinnate or the tripinnate leaves. This is the important feature. Then stamens are indefinitely long and prominent. So these two are the key features which I wanted to highlight. And these are the members in which are present in Mimosaidae. That is Acacia, Albizia, Mimosa, prosopis. Okay, so these are the things that I wanted to discuss. Then the second subfamily is the Caesalponidae. In the Caesalponidae, the important point is that the flowers show distinct imbricate estivation, that is ascending imbricate type. Okay, so this ascending imbricate type is important. Then pollen shed as monads, these are also important. Then the genera that are present are Bohemia, Delonix, Cassia, Caesalpinia. Okay, so these are some of the important specimens or the examples that are present in Caesalpinoideae subfamily. Now, the third to talk about is the last subfamily is the Papilionoideae, and in this, the important point is descending imbricate, also called as vexillary. That is, the posterior petal is outermost and largest, and it is also called as banner. Okay, then keel is also important. What does that mean? That two basal petals are ponate, and they're together called as keel. The two remainings are called as wings. So you should be knowing about this. Then the genera in which are present in this are Pisum sativum, that is mutton. Okay, Lathyrus, Physiolus, Lupinus, Arachis, or these are the example of Papilionoideae family. Then we move on to the next family that is important, and that is Cucurbitaceae family. The common name is cucurbit family, and in this the tendrils born on the nodes. So this is important point to remember. And then the leaves are simple, alternate, and they are palmly lobed, and they have got teeth of cucurbit type. So there is a presence of teeth, and that is important feature. Then we talk about the filaments that is present in the anthers are fused, and such condition is called synandrous condition, which is very important. You should be knowing what is synandrous condition. Then the ovary is inferior, and the placentation is parietal. Sometimes they ask about placentation from the cucurbit family. So if whatever I have done in the bold, that you should remember. Although you should go through all this so that it is easier for you to remember those points, and then just solve the question in the exam. Then we move on to the another family, and that is Apiaceae or the carrot family, and it is also called as umbelliferae, which is very important. This was an old name of it. And these are the herbs, but these are aromatic plants, and they are with hollow stem, and the stem is hollow in the internodal section. So this is an important feature to talk about. Then inflorescence is characteristic, and which is the umbel. So inflorescence is very important in case of Apiaceae. Then ovary is inferior, and placentation is exile. These kind of questions they tend to ask. Then they ask about fruit, which is schizocarp or the cremocarp. And the stylopodium is also important. The style that is swollen from the base and is modified as a nectar, and the secreting structure is called as stylopodium. This is also important. I've seen a question in the older part from here. Okay, so then we talk about the sixth family, and that is Rubiaceae family or the coffee family, and the common name is coffee family. The leaves are this point is important, that the leaves are opposite to the whorl and stipule leaf like interstipulate. Okay, so this point is important. Then the collectors, that is the multicellular secretory hairs, are present on the adaxial surfaces. This is also important. Then flowers are bisexual. Heterostylous condition is important for you to remember. Then the stamens are 4 to 5 and they are epipetalous. So epipetalous condition is seen in this, and the ovary is inferior. So these are the key points that you should remember. Then you should also remember the this family is economically important. Why? Because it provides Coffea arabica, and this is important. So you should be knowing about it. Then red dye comes from Rubia tinctoria. So let us talk about the seventh family, important family, and that is Asteraceae, which is also called as sunflower family. The old name is Compositae, and it has got the members which are shrubs, herbs, or trees, and they have got resin canals, which is very important. With the laticifers are also present in the resin canal, so that is important. Then sesquiterpene lactones is present inside this laticifers. This is also important. Then the inflorescence is capitulum. Sometimes they tend to ask in which of the following you'll find inflorescence capitulum. So you should be knowing that Asteraceae has got the inflorescence which is capitulum. These kind of question you can expect in exam. Then involucre of the bracts are called as phyllaries, and flowers are florets, and they are highly reduced. This is also a very important feature of Asteraceae family. Then flowers are bisexual or unisexual. Sometimes they are sterile or radial or bilateral. Then pappus is also important. Sepals are highly reduced, forming characteristic hair-like appendages, which is called as pappus. Then disc floret is also important. Then 5 petals forming a tubular corolla. Generally, they form a disc floret. So disc floret is seen in the case of Asteraceae family. Then we talk about ligulate, and in the ligulate we talk about the bilateral corolla, which is formed by 1 or 2 upper lips and 3 to 4 lower lips. Okay, so 1 or 2 upper lips and 3 and 4 lower lips together form the ligulate. So this is also important. The most important part I've told you about the capitulum, about this presence of resin canals and the laticifers and pappus. Okay, so these are the important parts that you should be knowing. And the older name is also very important, that it was earlier known as Compositae. So this was the seventh. Now the eighth one, this is Asclepiaceae family or the milkweed family. You should be knowing the name. Why it is called milkweed family? Because it contains milky latex in the lattice. So whenever you break that stem, you'll find milky latex coming out. So this is the identification feature of this family, and this is very important. And the collators may be present in this, and the corolla allows the appendiculate structure, which is called as corona. So this is also important, and because of the disease this has become important. So just try to have a look at then. Then the important term that I wanted to introduce you to is the gynostegium. Okay, so what happens in this, that the androecium and gynoecium are fused together and they form a structure which is called as gynostegium. Now, fruit is also important, pair of follicle or iterio or two of two. Okay, so this is important. You just have to, you should be having this information. You can just strike the wrong ones out and take the or pick up the right one. Now, that was the eighth one. Let us now talk about the ninth important family, and that is the Apocynaceae or the dogbane family. So this is the common name of this family, and here the laticifers are present. This is an important point. Then corona is also present, and gynostegium is also present, that we have just discussed in the last family. Then the point about the pollen grain is so important, that the pollen grains are tricolpate. This is very important. Then they form a sticky mass which is called as pollinia. Sometimes they ask in which of the following you'll find pollinia. Pollinia is found in Apocynaceae family. So this is an identification feature. And pollinium diagram you can see over here. And then we talk about nectar glands in this, and the fruit is a trio of two, like just like the previous family that we have discussed. Now let us move on to the next family, and that is the 10th family, I guess. That is Convolvulaceae family. And in the Convolvulaceae family, which is commonly called as morning glory family, and it is rhizomatous. And this is very important, that which of out of which of the following that is rhizomatous. So this is rhizomatous, and laticephers are also found in them, and it also contains milky sap in it. And there may be, in addition to it, there may be milk alkaloid may also be present with the milky sap. Then we talk about sepals are five, petals are five, they are gamopetalous, and they are funnel shaped, and they are distinctly plicate with many folds, and corolla is convolvulate. Okay, so convolute होते हैं, and this is an important feature that you should be knowing. And this family is important. Just revise this family. Convolvulus cuscuta are the representative of this family. Then we talk about the next family, that is Solanaceae, or the potato family. In this, the leaves are exstipulate with oblique leaf base. Oblique leaf base is an important identification feature which is present. Then we talk about oblique septum with the swollen placenta inside the carpel. Okay, so this is also an important feature, and the important members are Solanum, Datura, and this family becomes Petunia. This family becomes important because of the Solanum. Okay, so placentation here you can see, and these oblique septum is there with swollen placenta. Okay, so that is important. And placentation से question आया हुआ है, but थोड़ा पुराना है, but there was one question I've seen. Now let us talk about Lamia family, or which is commonly called as mint family. Okay, so this was 12. Now this has got a stem which is quadrangular stem, and if we cut out TS of that stem, we will find phenolic compounds. Then trichomes are also present. Trichomes are glandular with ethereal oils. Then we talk about inflorescence, which is racemes, and or verticillaster. Okay, so this is the second name for the racemes, or the verticillaster. So that is important. Then dicaseal cyme with monocaseal branching. That is also important, that we see inside the flower. Just go through this, and that is important. And then this is very important, that I wanted to tell you, that the stamens are four and they are didynemous. So this is an important feature. This is an identification feature of this. And then the style is gynobasic. This is also important. This is present in salvia exclusively. So that's why it is mentioned placentation is exile. So these are the few things that I wanted to tell you about, and go through these family. These are very important. Then the last one, I guess this is the last one. Uh, the one एक और है इसके बाद. Then Euphorbiaceae family, which is commonly called as spurge family. Now in this you have to see is that the leaves are sometimes stipulate, and they contain nectar glands at the base of the blade or on the petiole. So the presence of nectar gland is important. Then the false flowers that are formed, and that they are called as cythium. So this is an important feature which have been highlighted in the black. Then the last is the fruit is a shizoka. So that is the three or four features are important, but I have given you all the botanical features that are present inside the family. Don't miss out on the things that I have highlighted, that are important, that are in bold. Don't miss them. Okay. Then we come on to the Poaceae family, or the grass family, and the important point is that the leaves are alternate, and they consist of sheath, blade, and a ligule. So ligule is important, and ligule is present on the adaxial surface of the sheath. That is important. Then we talk about inflorescence. Inflorescence is compound or branched spike, or panicle, or spike of spikelet. So this is inflorescence part is important in the case of Poaceae. Then we talk about gloom, lemma, palea, on. So this is important. Go through them. Then they might ask you some question from the scutellum. So you should be knowing what is a scutellum. That is part of endosperm. Okay. Now in fungi, let us discuss the first phylum, that is Zygomycota. You know that the asexual reproduction in the members of Zygomycota takes place by the formation of sporangiospores inside sporangium on the stalk-like structure which is called as sporangiospore. Now in the order Entomophorales, there is production of conidia. So this is an exception. From here, the question can be made. Now, sporangium contains a sterile tissue which is called as columella. This is also a peculiar feature. Few members contains or makes sporangiola, which is a miniature form of sporangium. Then what is merosporangia and the fungus shotgun, which is seen in Pilobolus. So these are the different features which are important for the exam. Whereas there are different kinds of methods through which vegetative reproduction is seen, whereas sexual reproduction takes place, and this sexual reproduction takes place by the effect or the role of hormones. The sexual spore that is formed is called as zygospore, from which the name of the phylum is derived. Now, the type genus Rhizopus is quite important. Rhizomus common name is called bread mold. The cell wall composition you should be knowing, the mycelium that it contains, and the mycelium is formed by the hypha. The hypha has got three stages, or there are three kinds of hypha that you should be knowing. The economic importance of Rhizopus is also important. I have mentioned one, Rhizopus or rhiza, which is used in the production of alcohol through alcoholic fermentation. Now let us discuss the phylum Oomycota. Now, this phylum is different from the true fungi. So, what are the different features that makes it different from the true fungi? Okay. Then the asexual reproduction takes place by the formation of biflagellate zoospore, which is a difference from the true fungi. The main thallus is the diploid thallus. That's why the meiosis takes place in the developing gametangio. Then what are the sexual spores? These are these are the oospores. The sexual spores are the resting spores, and they have got a thick wall around themselves. Cell wall is composed of beta glucan, hydroxyproline, and cellulose, unlike chitin. Now, chitin is also found, but it is rare. It is found in Achila and Saprolegum. The mitotic and meiotic divisions are intranuclear and centric. This is also difference. Then it causes disease in mammals, which is called as pitheosis, and the causal organism is Pithiosum insidiosum. The terrestrial forms are parasitic forms. They cause plant pathology diseases in agronomically important crops. Now, Phytophthora infestans causes late blight of potatoes, whereas Plasmophora viticola causes downy mildew of grapes. Phytophthora cinnamomi causes or infects more than 1,000 species of plant. Now, the thallus, it can be holocarpic, eucarpic. You should be knowing these terms. Then the haustoria is used for what? And what are the different shapes of haustoria? You should be knowing that. The tip of the fungal hyphae contains apical vesicle, which has got cytoskeletal system. What is the significance of it? Then it lacks spins and copper, which acts as supply center into fungi. So what is the role of this, which is absent in this? And the mitochondria is with tubular cristae, whereas which kind of cristae are found in the true fungi, and Golgi apparatus in the Oomycota is multiple flattened cisternae. Then what kind of Golgi bodies are found in true fungi? You should be knowing the differences. Then fingerprint vacuoles. What are these fingerprint vacuoles? And they contain mycolaminarin and mycolaminarin phosphate. These are involved in the formation of a vacuole inside the vacuole of the egg cell or the oosphere, and which is called as ooplast. Then you should be knowing about the asexual reproduction. It is by heterocont zoospore. What are heterocont zoospores? And these sporangias, why do they resemble conidias? Because they are produced in chains. Then biflagellate zoospores can be of two types, that is primary and zoospores and secondary zoospore. You should be knowing the type of both the kinds, and what are the different organelles that contain, as well as what are K bodies. So these kind of details you should be knowing about these zoospores. Then we move on to the basic biochemical differences between the true fungi and oomycetes, like the synthesis of lysine uh, takes place via alpha amino adipic acid pathway in true fungi, where it is diaminopamelic acid pathway in oomycetes. Ergosterol hota hai sterol, and whereas sterol is fucosterol in case of oomycetes. Glycogen is the reserved food material, whereas it is mycolaminar. Sugar alcohols are present in true fungi, whereas it is absent in the oomycetes. Then we move on to the type study, and that is for the first one is Phytophthora. It causes Phytophthora infestans causes late blight of potato in Solanum tuberosum. You should be knowing the host plant, the causal organisms, as well as the disease name. So these three things you should be knowing. The other species are Phytophthora erici, which causes diseases known as cholera, or it causes disease of erica palms. Then Phytophthora colocaceae causes blight of colocasia. Phytophthora parasitica causes seedling blight of castor, as well as leaf and foot rot disease of palm. Phytophthora palmivula causes bud rot of toddy palm. Okay, so you should be knowing this. Apart from this, it also causes bud rot of coconut palm. Then what are the control measures for Phytophthora infestans? Then the use of disease-resistant tubers, resistant varieties, then dipping the tubers in mercury chloride, or the use of fungicides like Blitox, Phytolan, Dithane Z78, Dithane M22, or body mixture. Then we move on to the second genus of the phylum, and that is albugo. It causes white rust of crucifer in the crucifery family, and albugo ipome panpandurini causes sweet or it attacks sweet potato or convolvulaceae family. Albugo potulici attacks the potulica, or albugo bletae attacks the Amaranthaceae family. Then the tissue shows hypertrophy. Then what is hypertrophy? What is hyperplasia? And we see swelling and distortion in floral parts. In the case of white rust of crucifer, then how it is formed and what is responsible for it. The hypertrophied parts of the plant contains the oospores of the fungus. Then what are the control measures like clean cultivations, crop rotations, or spraying the infected plants with the copper fungicides? So this is a picture that shows you sporangiospores of Perennosporosi family, which also comes under the phylum Omycota. Now let us talk about the next phylum, and that is Ascomycota, which is also called as sac fungi. Because sometimes they ask questions from the common names in the last year paper. You can see that they asked about cup fungi, which is Phaziza. Okay, so ascospores are produced, which are the sexual spores, and how they are produced, in what manner, like they are produced in endogenously, whereas in Basidiomycota, they are basidiospores are produced exogenously. So this is the difference I should be knowing. Then how many ascospores are formed in an ascus, like 8, 4, or multiple of 8? Flagellated cells are altogether absent in the case of Ascomycota. Then what are coprophilous form of fungi which grows on dung? Then hypogene form kaun sa hota hai? Then what are the two types of placenkyma, like prosenchyma? What is prosenchyma? And then what is pseudoparenchyma? So you should be knowing these terms. The cell wall is chiefly chitin, and the diseases that the members of Ascomycota causes, like ear rot of corn, scab and foot rot of cereals, brown rot of stone fruits, apple scab, peach leaf curl disease. Then we move on to asexual reproduction. The asexual reproduction is mainly through conidia. Then what are the conidiophore types? Like they can be branched, where it can be branched, like in penicillium. Unbranched का होंगे aspergillus. Then what is conidiogenous cells? Then these conidiogenous, what are the types of conidiogenous cells, like phyllites or the sterigmata or the annelids? Then these conidiophores may be present singly in the case of Ericyphe, whereas they may be forming conidiomata or the asexual fruiting bodies. Then what are the types of the asexual fruiting bodies, like Sporodocium? In which genus you will find them? Cynema. Example, you should be knowing Pycnidium, Acervulus. So you should be knowing these asexual fruiting bodies. Then we move on to the paired nuclei that are formed inside the ascus. Then you should be knowing how the ascus is formed, how meiosis takes place followed by the mitosis. What are the number of ascospores that are formed in the ascus, and the methods by which the two compatible nuclei are brought together so that there will be formation of dikaryon or dikaryotic hyphae. So the gametangial copulation, gametangial contact, hologamy, spermatization, somatogamy, you should be knowing these terms. Or you can expect question from here. Then development of ascus, whether it can be indirect and the direct ascus. Then you should also be knowing about procedure development. What is penultimate cell? What is synkaryon? Then in Trichobolus ascus, there are around 700 ascospores. This is also quite important. Then the picture shows you different kind of sexual fruiting bodies. The ascospores are formed in the ascus, and these ascus can be scattered or naked, as in hemiascomycetes, whereas it can be formed in a enclosed in a fruiting body which is called as ascocarp. And what is hymenium? Hymenium is a layer which contains the paraphysis and the ascus. So, what are the different kinds of ascocarp like Cleistothecium, Perithecium, Apothecium, and Ascostroma? So, last year there was a question from this Apothecium cup-shaped fungi. So, you should be knowing these terms, and you should also be knowing the different genuses in which they are present. Then we move on to the type genus of Ascomycota, and that is Aspergillus. Aspergillus it mainly shows only the asexual reproduction, and it is through unbranched conidiophores. The conidiophores can be uniseriate or biseriate. Then what are the different parts of these? You should be knowing the diagram. Then sexual fruiting body, if sexual reproduction is found, then it is Cleistothecium. Most of the members do not show sexual reproduction. You should be knowing the economic importance of Aspergillus, like it causes a disease called aspergillosis. What is that disease? Automyces it causes in humans. Apart from this, Aspergillus oryzae is used for wine production. Aspergillus niger is used for biosis or for the detection of copper. Then Aspergillus gossypi is used for the production of vitamin B. Antibiotics like flavicin and spergillin are extracted. So these are the economic importance you should be knowing. The second genus is the Penicillium, and in this also asexual reproduction is mostly seen, and it is through branched conidiophore. Sexual fruiting body, if found, like in this species, it is Cleistothecium. Most of the members lack sexual stage. The economic importance you should be knowing what are the different economic importances that are related. Like the antibiotics, penicillin is extracted from P. notatum and Penicillium chrysogenum. So you should be knowing these terms. As moreover, you should also be knowing some other like the pigments. What are the different pigments that are extracted from penicillium, like citrulline, spinulosin, and amodic acid. So just have a look at the economic importance. Then we move on to the third type genus, and that is Claviceps. It causes Claviceps purpurea causes ergot of rye in the rye that is sickle cereal. So this is the host plant. You should be knowing three things: causal organisms, host plant, and the disease name. Then the other species is Microcephala. It causes ergot of bajra or ergot of sorghum. Then consuming ergot may cause problems like vomiting or heat and cold. A person may feel heat and cold. Then the next type genus is Pizziza, from which the question was asked last year. Asexual reproduction is through conidia. Sexual fruiting body is apothecia. Most of the members lack sex organs, and the compatible nuclei are brought together by somatogamy. Then yeast, the next type genus is yeast, and the asexual reproduction is through budding and fission, as you already know. Sexual fruiting body is naked acile, and the process through which nuclei are put together is hologamy. Alcoholic fermentation is done by the yeast, and the two enzymes that are involved are zymase and invertase. It is a good source of vitamin B. Then we move on to the next class, that which is called as a form class. It includes all the imperfect fungi or the asexual Ascomycota. And sexually reproducing fungi, as you know, are called as perfect fungi. And the classification of fungi is based on what sexual spores like oospores, zygospores, ascospores, and basidiospores. This is an artificial group. So all the fungi which do not show sexual form are included in them. What are the different members that are included in them? Then what is the term which is known as pleomorphy? What are the terms that important terms that you should be knowing are synamorphs, क्या होते हैं, holomorphs, telomorph, anamorph, orphan anamorph. So you should be knowing these terms. Then why there is loss of sexuality? It is through mutation, hybridization, etc., etc. You should be knowing these terms, as well as why there is no sexual reproduction reported. And what is the if the sexual reproduction is not happening, then what is the source of variation? Then Alternaria, it causes early blight of potato, which is included in this form class, and it is caused by Alternaria solani. And Alternaria tenuis causes black point disease. Then Alternaria brassicae and Alternaria brassicicola causes leaf spot of crucifer. Then what is target board effect that we see in the case of early blight of potato? Then we move on to the next phylum, and that is Basidiomycota. It includes mushroom, toadstool, puffballs, rust, and the smuts. Then the spores that are produced, what are the kinds of spores that are produced? That is non-motile spores are produced. What is basidium? What is the shape of basidium? What happens inside the basidium, like karyogamy and meiosis happens? And basidium produces four kinds of four spores which are called as basidiospores exogenously. This is different from ascos or the ascospores. Now, sexual spores, these are the basidiospores. Then what is rhizomorph that we talk about in Basidiomycota? Then mycelium passes through three stages: primary, secondary, and tertiary mycelium. So you should be knowing the characteristics of these mycelium. Then what is dikaryoticization or diplodization? How this dikaryoticization is reached or dikaryotic condition is reached. Then what is clamp connection? How they are maintained. Then what are basidiocarps or the fruiting bodies or the sporophores, which are produced by secondary mycelium. And the basidiocarp corresponds to what? Ascocarp. The rust and the smust do not produce any basidiocarps. The size of the basidiocarp is variable. It can be very minute to more than 3 feet. Then what is basidium? How it is developed? What are the types of basidium, like holobasidium or phragmobasidium? And what are the condition of septation, whether it is septate or non-septate? And if it is a phragmobasidium, what are the types of phragmobasidium? Stichobasidial, what are the genuses in which you will find stichobasidial like Astilago, or Chiastobasidial or tunic fork? So you should be knowing the characteristics as well as the example in which they are present. Basidiophores, wall layers you should be knowing. Then what is water drop mechanism? Then in this table, you'll find the key differences between the basidiomycetes and the ascomycetes. Like in basidiomycetes, it is dikaryotic phase, is very long-lived and independent, whereas it is short-lived and dependent. The formation of basidia and basidiospore, here it is asi and ascospores are formed. Basidiospores are formed exogenously, whereas it is formed in endogenously inside the ascus. Four basidiospores are formed, eight ascospores are formed. Fructifications of basidiocarps, here it is called ascocarps. Common feature is clamp connection, here it is crozier formation. Sex organs are mostly lacking, it is present only in the primitive members. Then we move on to the type genus, which is called as Puccinia, which is very important. It derives its name from an Italian scientist, which is called as T. puccinii. It is also called as rust. Why it is called as rust? Then wheat is attacked by three rust fungus, and one is black or stem rust, which is caused by Puccinia graminis tritici. Yellow or stipe rust, which is caused by Puccinia striformis. Then brown rust is caused by Puccinia recondita. Then what is a heterocious fungus? Then what is macrocyclic or polymorphic fungus? Rust or the fungus you should be knowing. Lifespan of Puccinia is around 1 year. Then how many spores it forms in its life cycle? What is primary host? What is alternative host or the secondary host? The different kinds of spores that you'll see in the case of Puccinia are iridospores, Teluto or the teliospores, Basidiospores, Pycnidiospores or the Spermatia, or Acidiospores. These iridospores germinate on primary host, whereas Telutospores or teliospores germinate on primary host. Basidiospores germinate on the ground. Pycnidiospores or Spermatia germinate on alternative host, that is Barbary. Acidiospores are germinate on alternative host. Then these are the stages based on the different kinds of spores that are produced: irido stage, teleutous stage, basidial stage, pycnidial or the spermogonial stage, or the acidial stage. Then what are the control measures? How we can control the spread of this Puccinia? That is eradication of alternative host, that is Barbary, or dusting with diethane, cyclohexamide derivatives, or Zinane. Then early sowing of wheat, because it infects at a particular stage of wheat. Then cultivation of resistant varieties of the plants or the host can also control the spread of Puccinia. Then we move on to the next type genus, which is called as Astilago. Astilagos are called as smuts. And what give this name smuts to them? Then Astilago maydis causes smut of maize. Astilago avini causes loose smut of oats. Astilago nuda causes loose smut of barley. Astilago tritiki causes loose smut of wheat. Astilago hodai causes covered smut of barley. Astilago skeetamani causes whip smut of sugarcane. Astilago choleri causes covered smut of oats. So these are kinds of diseases that are spread by the Astilago. Then what are the control measures like hot water or solar treatment or use of resistant varieties like NP710, NP718 or 770, Bansi Pali 808, Bansi 2240, or the use of fungicide. So these are the different methods by which what is controlled. Uh, Black colored spores or the smut of wheat is loose smut of wheat is controlled. Okay, so these are the control measures for loose smut of wheat. Then we talk about covered smut of barley, which is caused by Astilago hodai. It occurs mainly in the regions of UP, Madhya Pradesh, Bihar, and Punjab. Then you will see the manifestation of disease as blackened ears, and there is a presence or the formation of intercalary teleutospores, which is very important. Then we will discuss. You should be knowing the differences between the rust and the smuts. That smuts are the obligate parasites, whereas the smuts are not obligate parasites. They can be heteroecious or rotaceous, but smuts are always rotaceous. Clam connections are less. Here, clam connections are very common. Teleutospores are terminal, where it is intercalary. Teleutospores can be two-celled, and each cell has got two nuclei, whereas teleutospores of smuts is unicellular and binucleate. Basidiospores are produced on sterigmata, and the number of basidiospores are four, whereas number of basidiospores are not definite and they are not produced on sterigmata. And basidiospores are discharged violently by water drop mechanism, whereas in smuts there is no such mechanism and they are not discharged violently. Then we move on to the another type genus which is called Agaricus. It is also called as Silota. Agaricus campestris is the best edible disease. Then what are the poisonous mushrooms called as? Which is called it is they are known by toadstool name. Amanita muscaria, Amanita varna, and phylloides are the deadly poisonous species of mushroom or the Agaricus or the mushroom. Then which kind of mycelium, whether it is primary or secondary, which is found below the ground or beneath the ground? Then where the fruiting bodies or the basidiocarps are found? Then if we are talking about basidiocarp, then what are the parts of basidiocarp, like stipe? What is a stipe? What is pileus? What is annulus? And how is inium arranged? What are gills? Then what are fairy rings? Then what is heterokaryosis? Parasexuality. So you should be knowing this. Who discovered parasexuality? Where, in which genus it was first discovered? What is parasexual cycle? What is alternative to? Now, as you know, the members of the algae show different life cycles. Now, there are three major life cycles that are seen. That is the haplontic life cycle, which is shown by Chlamydomonas. The other type of life cycle is the diplontic life cycle, which is shown by Cladophora or Acetabularia. The third kind, which is shown by maximum members of the algae, is the isomorphic alternation of generation, where both the haploid and the diploid phase are equally represented. Then Effie Frisch is regarded as international father of the algae, whereas MOP Iyengar is regarded as Indian father of algae. Now, Effie Frisch divided the algae into 11 main classes, and you should know these 11 main classes, the pigments that they contain, the color of the chromatophores that reflects, the reserved food material that each class contains, the composition of the cell wall, as well as the genera that these classes contain. So these are the four major fields that you should be knowing about. We will be discussing few classes, and the rest you can see from the table. The first one to discuss is chlorophyce, where the reserved food material is a starch, the cell wall is cellulosic, and the examples are chlamydomonas and bollworms. Let us talk about the pigment. The pigments are chlorophyll AB, as well as carotenoids, which is yellow pigment. Now, chromatophores are grass green in color. Now let us talk about the second class, which is the xanthophyceae. Now xanthophyceae contains reserve food material as oil, and the cell wall is rich in pactic substances. Whereas the examples or the representatives are Vaucheria and Botrydgium. Talking about pigments, then the pigment is mainly xanthophyll, which is yellow, and the chromatophores are yellow-green in color. Now coming on to the third class, important class, that is Phaeophyceae. And in Phaeophyceae, the reserved food material is present as alcohol, as mannitol in the form of alcohol in the form of mannitol, then polysaccharide in the form of laminarin, and fats. As well as there is the cell wall is made up of cellulose, and the various representatives are ectocarpus and fucus. It also contains fucoxanthin as the pigment, and chromatophores are brown in color. Coming on to the second last class, and that is the Rhodophyceae. Now, Rhodophyceae contains the reserved food material as the Floridian starch. The cell wall is cellulosic in nature, as well as the examples are the polysiphon. The pigment that it contains is phycoerythrin and phycocyanin. The chromatophores are red, bluish in color. Coming on to the last class, which is Cyanophyceae, and it contains a reserved food material as sugars and glycogen. The cell wall is mainly composed of mucopolymers, as well as the examples are acetoluria, anabina, nosto. The chromatophores are blue-green in color. The chlorophyll, uh, carotenes, xanthophylls, phycocyanin, and phycoerythrins are the pigment. Now, talking about this cyanophyceae, it contains nostoc, and these cyanobacteria belong to the order Nostocales, and they produce three kinds of cells in their life cycle. That is vegetative cell, heterocyst, and spores. These heterospores are of different shape. The main function of heterocyst is to fix the nitrogen. So this is important. Volvox is also called as rolling alga, and beside this, there is one alga which is called as chlorella. Now, chlorella is an important alga because it has got some economic importance. Now, this has got a very high protein content, around 50% of the protein content it is found, 20% fats, and 20% carbohydrates. It is also a source of riboflavin, vitamin A, vitamin B12, choline, and it is used as consumption of human food. Then it is also used as a space food, and chlorella gives out antibiotic chlorelin, which works against bacteria. Now, this it has got a capability that it can remove CO2 from water. Therefore, it is used as biological filter in water reservoirs. Now, coming on to then another important alga, and that is Spirogyra. In Spirogyra, uh, sexual reproduction is important, but before that, it is also called as pond scum or pond silk. Now, sexual reproduction is isogamous type of sexual reproduction, and it occurs by the conjugation of non-flagellated gametes. Two types of conjugation is seen. One is scalary form, and the other one is lateral. So, conjugation tubes are formed. Then, bioluminescence is also observed. Bioluminescence is observed in nocticula. That is phenomenon of emission of light. It is seen in dinoflagellates. Then the last one to tell you about is the oldest fossil. It is in fossil diatoms, and that deposit of fossil form is known as Kieselguhr. The diatom responsible for it is Pixidicula. So you should be knowing about these terms, and they are important for the exams. Now coming on to the type genus, let us talk about the first member of the Chlorophyceae, and that is Chlamydomonas. In this, the most important species is Chlamydomonas nivalis, and this is also called as red snow because it contains hematocrit. Now, this Chlamydomonas contains chloroplast, and this chloroplast can be present in different shapes. Okay, so one question has been asked from net-like chloroplast or net-shaped chloroplast is present in which of the species? The answer was Chlamydomonas reticulata. So they can ask different shapes of these chloroplasts are present in different species of Chlamydomonas. So I have already put it here. You can just have a look. Then you should be also knowing the structure of the thallus and what are the organelles that it contains. The two main important organelles is the stigma and the pyrenoids. What are their function? What is the role? Now, if you talk about the stigma, then it is the organ which is called as a shading device, or it is meant for photoreception. And now, if we talk about its structure, then it has got a lens which is biconvex, and it is hyaline, and the lower part is the curved plate or the pigmentosa. You should be knowing about it. Apart from this, there is one flagellar apparatus that helps this that provides motility to this chlamydomonas, and it composes of paradesmos, and that lies between the two blepharoplast, whereas rhizoplast lies between the nucleolus and one of the blepharoplast. Now, talking about reproduction in chlamydomonas, then Pamela stage is very important in asexual reproduction. Pamela stage is observed in Chlamydomonas clani, and aplanospores are found in a common mucilaginous sheath. Then, talking about sexual reproduction, all the three kinds of sexual reproduction is observed. That is isogamous, anisogamous, and oogamous. The different species of Chlamydomonas that shows up these three kinds of reproduction is already mentioned, that you should know. And lastly, let us talk about the life cycle of this Chlamydomonas. Now, in this life cycle, you can see that in the sexual reproduction, there is shown that the isogamous type of sexual reproduction, and it leads to the formation of quadriflagellate zygote. Okay, so that is important. And in the asexual reproduction, Pamela stage is seen. So let us now move on to the next genre. The next genre is the hydrodictyon. In hydrodictyon life cycle, two things are important. That is the formation of daughter synovium and the formation of polyeder. These two things you should know from hydrodictyon. Now moving on to volvox. Volvox is also shows you synovium, and in volvox you should know how the gonidiums are formed, and how the inversion process takes place. Apart from this, you should also know how the oospore is formed. This is important with exam point of view. Now coming on to the next genre, and that is Chara. In Chara, the most important part is the formation of globule and nucleus, and globule and nucleus contains the sex organs. Globule contains the antheridium, whereas nucleus contains the oogonium. You can have a look at the life cycle also, where the Chara plant forms globule and nucleus. Inside it, antheridium and oogonium is present, which are the sex organs, and then anthrozoids and ovum are formed, which fertilize together, which fuse together to form a zygote, and then a resting spore is formed, which is called as zoospore, in which reductional division occurs, and then protonema occurs. Okay, so then this new Chara plant is formed. This is how it goes about. Asexual reproduction or vegetative reproduction is very important. That is, amylum presents the production of amylum stars and bulbil. Okay. So this happens in Chara. Let us move further ahead. Now in Oedogonium, which is also a part of Chlorophyceae, which belongs to Chlorophyceae, two forms are seen. One is Macrandrous form, and the other one is Nenandrous form. In Nenandrous form, dwarf males are formed. So what are the difference between the Macrandrous form and Nenandrous form, and what are the basic significance of both? Okay, so you should be knowing that. Apart from this, you should also know about the capsule, the vegetative structure of Oedogonium cell, and the presence of capsule, how the capsules are related to the cell division. You should be knowing that number of capsules are equal to the number of division it has undergone. Okay, so these are the few details that you should be familiarized with. Very similar to Oedogonium is the Eulothrix, and in Eulothrix, you should know the basic structure or the vegetative structure that I've already drawn, and the individual cells are placed one above the other. The basal part contains the hapron or the rhizoidal cells on the or the holdfast. Below it, I have mentioned few questions like what is the mode of sexual reproduction in Chlorophyceae. Then all of the three methods are seen in the Chlorophyceae. If we talk about Eulothrix, produces what kind of spores? Then it produces isogametes. So both the gametes are of equal size. Then the last question is, which of the following has non-flagellated isogametes? Then it is the Spirogyra. Through conjugation, they move from one thallus to another. So let us move ahead. Now we talk about the next class, that is Xanthophyceae, and it is about Vaucheria. In Vaucheria, you can see that there is production of zoospore, aplanospore, icanids in the vegetative reproduction, and simple sexual reproduction takes place that leads to the formation of oospore, which undergoes reductional division. Okay, then we come on to the next class, that is Phaeophyceae. In Phaeophyceae, there are two, Ectocarpus and Fucus. First, we are discussing Ectocarpus, and you should know that in Phaeophyceae, the main plant body is diploid, and in Xanthophyceae as well as in Chlorophyceae, we have seen the main plant body was haploid. Now, in the case of Ectocarpus, you can see that the sporophyte is diploid. It forms unilocular sporangium. reductional division takes place, and then the unilocular zoospores or the meiospores are formed, which will give rise to gametophyte, which will be haploid. Then on this gametophyte, what will be developed? Plurilocular gametangia will be developed, and it will produce isogametes, which will be haploid in nature. These two isogametes will fuse and they will fertilize to form a zygote. Then there will be reduplication of the sporophyte that was earlier present. Now this sporophyte can also form plurilocular sporangium, and then plurilocular sporangium will form a neutral sporangium, and in the neutral sporangium, neutral spores will be produced through mitosis, and that will give rise to new plants. So this is reduplication of the sporophyte. Otherwise, it will undergo sexual reproduction to form this gametophyte. So this is how alternation of generation takes place. It is quite different from the different members of the algae. Now let us talk about the fucus. In fucus, the main point for you to remember is the formation of receptacle. And in receptacle, there are male conceptacles and female conceptacle. Male conceptacle will have antheridium. Female conceptacle will have oogonium. And antheridium will form anthrozoid. Oogonium will form eggs. Then the same life cycle goes above. So this was important regarding the fucus. The last that is Polysiphonia belongs to the class Rhodophyceae, in which male and female gametophytic plants are different plants. So heterothallus is seen, and the both the plants will form the respective sex organ, that is the spermatangia, the male sex organ, and the carpogonium, that is the female sex organ. It will produce a spermatia or the egg. Then it will lead to the formation of zygote. Then this zygote will develop and form carposporophyte, which will be diploid. This carposporophyte will form carposporangium, and this carposporangium will have the carpospores produced mitotically. These carpospores will be diploid, and they will germinate to form tetrasporophyte. These tetrasporophyte will produce again tetrasporangium, and inside the tetrasporangium, reductional division will occur to form tetraspore. Now the final spores will be the haploid spores. This will again give rise to gametophytic plants, and these spores will be both male female types, so both kinds of plants will be formed. You have to remember that gametophytic male plant, gametophytic female plant, one form of plant. The other form of plant is the carposporophyte, and the third form of plant is the tetrasporophyte. So two diploid forms of the plant, whereas one gametophytic or the haploid form of the plant. That we about bryophytes. Now, in the bryophytes, the classification is important, and the three main classes in which the whole bryophytes are divided are the Hepaticopsida, Anthocerotopsida, Bryopsida. The Hepaticopsida contains several orders, and you should know Rixia and Markentia comes under this class. Then the class Anthocerotopsida includes the genus Anthoceros. The Bryopsida includes the sphagnum, Paleoporella, and the Phinarium. Now, these are the different genera that are present in different classes. So you should have a rough idea about the classification. Now, let us talk about the generalized life cycle of the Rixia, that is the type genus. Now, in Rixia, the gametophytic plant, the main plant body is what? Haploid. It produces the sex organs antheridium and archegonium on the plant body, on the same plant body, and as well as on the different plant body. Now, when these antheridium and archegoniums are produced, they produce gametes such as anthrozoids and the egg, and they fertilize to produce the zygote, and this zygote will form the sporogonium or the sporophyte. This sporophyte will produce the spores, and these spores are produced by the reductional divisions of the spore mother cells. Now, these spores will germinate to form the gametophytic plant. The gametophytic plant reduplicates itself with the help of vegetative reproduction. Now, germination of spores is through the germ tubes. Now, you should know that in the case of bryophytes, all the sporophytes are important. The sporophyte that is formed in the case of Rixia does not have foot and seta, only the capsule part is there. So for the whole type genus of this bryophyte, you should be knowing the structure of the sporophyte of different genera. Let us talk about Markentia, which also belongs to the class Hepaticopsida as Rixia. Now, the main plant body is haploid, as is the case in the Rixia. This plant body produces a specialized gamma cups. Now, these gamma cups contains gamete, and you should know about the gamete and the gamma cups. Now, this is the asexual or the vegetative reproduction that takes place in Markentia. Now, the plant body has got male plant and a female plant. Now, these male plant and female plant will produce antheridium and archegonium, and they will form gametes which will fertilize to form zygote. These zygote will form embryo, and the embryo will pass through various stages like the 4-cell stage, the octant stage, or the 8-cell stage, and then the sporophyte will be produced. The sporophyte, inside the sporophyte, the spores are produced, and these spores are the haploid spores which are produced after meiosis. That these spores will germinate to form new plant body, which is also a haploid. Now you can see on the right hand side on this diagram, this is a diagram of the Markentia sporophyte of the Markentia. Now in this you can see elaters. What is the function of elaters? Then you should also know about the calyptra. What is this layer calyptra, and what is from where it has originated, which component of the parent plant has contributed to this. Then you should be knowing about the perigynium, the jacket, how many layered jacket is present. Then you should be knowing about that foot, seta, and capsule are the three main parts of this sporophyte. So you should be knowing about the sporophyte in detail about Markentia. Now let us talk about the next genus, that is Sporella. Now this Sporella has got the sporophyte that you can see here. Now, in this sporophyte, you can see different structures like perianth is a new layer, and which are the components that contributes to this layer. You should be knowing about the elaters that are present, the spore mother cells, how these spore mother cells are formed, whether they are being contributed by the endothecium or the amphithecium. So this is the thing that they ask usually in exam. And the foot, seta, and the capsule. The jacket in the case of Porella is made up of 2 to 3 layered, or is it 2 to 3 layered jacket of the capsule. Now, coming on to the life cycle part, then the life cycle part is quite similar to what we have just discussed before, that the gametophyte is the main plant body is the gametophyte, which is the haploid. It will produce the antheridium and the archegonium on the respective male and the female plant. And they will produce gametes which will fertilize to form zygote. The zygote represents the first cell of the sporophytic generation, and this will undergo divisions to form mitotic divisions to form embryo. This embryo will form sporophyte, and this sporophyte will be dependent on the gametophyte. And then this sporophyte will form spores through reductional division, and these spores will form the gametophytes. So this was about the Porella. Then coming on to the Palea, another type genus, that is Palea. In the Palea, the sporophyte structure is quite important because there is a presence of elatophore, which is a new kind of structure that helps in dispersal of the spores. The sterilization, although is increasing, that you can see that as we move from Rixia to the Finaria, the sterilization part is increasing, that the number of sterile part is increasing. Then you should also know about the fate of involucre, the calyptra, and the jacket. So basically, you should be well versed with all these sporophytes of all the genera as well as the life cycle part. Now, coming on to the life cycle, then fragmentation and adventitious branches is the method through which vegetatively the gametophytic plant reproduces. Then coming on to the sexual reproduction, the same way in which that we have discussed in Porella takes place in the Palea also, where antheridium and archegoniums are formed. They will form the respective gametes, which will fuse to form a zygote. Then zygote will divide mitotically to form an embryo. This embryo will form a sporophyte, which will be dependent on the gametophyte. And inside the sporophyte, spores will, spores will be produced after the reductional division. Now coming on to the next type genus, and that is the sphagnum. Now this is called as bog moss, peat moss, that we'll discuss later. But look at the sporophyte diagram. In the diagram, you can see the different shape of the sporophyte, where it also has got a false foot, or the foot is inserted into the vaginella, and the foot is bulbous, and how the spore sac shape is different. It has also got annulus and the operculum, and it has also got calyptra layer, and the columella, which is the sterile tissue, is increasing in mass as we are moving from genera to genera. So this is called sterilization. So sterilization is increasing. Coming on to the uh, uh, life cycle part of sphagnum, then the gametophyte vegetatively propagates through innovation method, which is a new method. You should be knowing about it. Then the gametophyte has got male and female branch separately on the main plant body, and the male branch produces antheridium, whereas the female branch produces the archegonium. Both produces the respective gametes, which will fuse to form zygote. Zygote is diploid, so it will mitotically divide to form embryo. Then embryo will form a dependent sporophyte. This sporophyte will be dependent on the gametophyte for nutrition and support, and then the reductional division inside the sporogonium takes place. In the case of the spore mother cells undergo reductional division to form spores. You should be knowing whether the archaeosporium is spore or the endosporium is formed. So you should be knowing these terms, and then spores are formed, haploid spores are formed, which will germinate to form protonema, and then this protonema will give rise to gametophyte. Amphithecium or archisporium. You should be knowing these terms because several questions are being framed from this. The next type genus is the Anthoceros. In Anthoceros, the sporophyte is quite elongated, and there are various phases or regions of the sporophyte that you should be aware of. And each that is at the apex, in the middle part, at the bottom part of the sporophyte, different kinds of structures are seen. And what is the fate of columella? It is from endothecium. So that's why I was asking you about in all the type genus, you should be knowing the fate of archaeosporium and the endothecium. Question may come from there. Okay, in the apex part of the sporophyte of Anthoceros, you'll see pseudoelaters and spores, and in between there is a midrib which is called as columella, and in the lower part you will not find any spores, only the pseudoelaters will be there. Okay, in the middle part you'll find the spores as well as the pseudoelaters, and involucre will be present at the basal region of the sporophyte. There will be foot which will be bulbous. Okay, and seta is quite elongated. Theta is inconspicuous, and it is not elongated. The capsule is very elongated structure. So you should be knowing about you should be thoroughly go through this sporophyte part of the Anthoceros. Let us quickly discuss the life cycle. So fragmentation tubers, persistent growing apices, aposporia, and gamma cups are seen as vegetative reproduction in the case of Anthoceros. The gametophytic plant body, which is the haploid body, forms the antheridium and archegonium, which are the sex organs, and they form gametes, and then gametes fuse to form zygote. This zygote is the first cell of the sporophytic generation. Sporophytic generation. This undergoes mitotic division to form embryo. This embryo will form a sporophyte. This sporophyte then inside this contains spore mother cells. These spore mother cells will undergo reductional division to form spores. These spores will form gametophytic plant body. So this was all about Anthoceros. Let us now talk about the next genus, which is called as Funaria. In Funaria, operculum teeth are present. Inner peristome teeth, outer peristome teeth. Operculum is the cap-like structure that covers that teeth. Okay, so dispersal mechanism is quite important. Then you should be knowing about columella. Then this spore sac is quite different. Air chambers are also there. Then you should be knowing about what is apophysis. So the structure of capsule in the case of Veneria is also important. You should be knowing about the peristome teeth, the inner rim that it contains, the inner peristome teeth and the outer peristome teeth. Okay, so you should be knowing all these things, and that is important. Let us talk about the life cycle of Funaria. Then the Funaria reproduces vegetatively through fragmentation, tubers, terminal buds, aposporia, and gamma cups. And the sexual reproduction is quite similar to what we have discussed. The only different part is that the spores will first form primary protonema before forming the main gametophytic plant body. Now, in the case of Polytrichum and Pogonatum, just look at the structure of the sporophyte, and that is important, as the Polytrichum has got a sporophyte in which the operculum is slightly tilted, whereas in the case of Pogonatum is quite straight, and this is the difference that is present. Apart from this, Pogonatum also contains a cap-like structure, which is called as calyptra, which covers this sporophyte. So just go through the sporophyte structure of Pogonitum and Polytrichum, and that will be important for the exam. You may expect a question from it. Let us talk about the economic importance of bryophytes, which is important. Now, peat formation takes place through in the sphagnum and which is also called as bog moss. Then the sphagnum is used in the horticulture, and it serves as a bed on which the moisture can be retained for a very longer time. This sphagnum can also be used as a packing material for sending various plant materials to different places. Then sphagnum is also used in the World War as surgical dressing. Then, if we talk about sphagnum, it can be used as fodder for the animals, and it can also be consumed by humans. It is used for making breads while the China was under famine conditions. And various bryophytes, they are together with the lichens, are used as pollution indicators, and index of atmospheric purity is based on the number and frequency and the resistant factors of the species of the bryophyte. So this place say if the bryophytes have gone, that means that is the indicator of the pollution of that particular uh, substance. Okay, so these are used as indicators. Now, copper moss is an example which serves an indicator of higher concentration of copper. Okay, Dryptodon and okay, Dryptogon is an example. Melicofera feria is also an example. So you should be knowing the indicator plants that tells us about the pollution that particular pollution that occurs in the environment. Then sesquiterpenoids are also extracted from the bryophytes, and Boxbomia himalayensis is responsible for it. So just have a look at the economic importance that is important. Then two terms, then that is apogamy and apospores are important. That is the formation of sporophyte directly from the gametophyte without the formation of gamete is called as apogamy. And if I talk about apospory, then that means the formation of gametophyte from the vegetative cells of the sporophyte without the formation of spores. So these are some important terms and the economic importance that may be required for your exams. So about pteridophytes. Now, as you know, pteridophytes can be grouped into two main groups, those producing single type of spores, which are called as homosporous pteridophytes, and the other kind is the heterosporous pteridophytes, which produces two kinds of spores. Okay, so the homosporous pteridophytes includes Lycopodium, Dryopteris, and Equisetum, whereas the heterosporous pteridophytes includes Slaginella, Marsilia, Isoetes, and Azula. What happens is when zygote is formed, the zygote forms the sporophyte which is diploid. It produces spores of equal shape and size, and these spores are produced after reductional division. So these spores are haploid. These spores give rise to a haploid gametophyte, which then forms the uh, the female and the male gamete. These female and the male gamete, that is egg and anthrozoid, fuse to form zygote again. So this is the generalized life cycle of a homosporous pteridophyte. Whereas if we talk about the heterosporous pteridophyte, that includes Slaginella, Marsilia, Isoetes, and Azolla, in this the zygote is formed. This zygote gives rise to sporophyte. This sporophyte is diploid. It will form two kinds of spores. The one kind of spores are smaller in size, and both the kind of spores are formed after reductional division. The other kind of spores are the larger in size, and these are called as megaspores. The smaller spores are called as microspores. Microspores forms the male gametophyte, whereas megaspores germinate to give rise to form the female gametophyte. These male gametophyte and female gametophytes are formed separately. The female gametophyte forms the egg, whereas the male gametophytes or the prothallus, which is called as the gametophyte is called prothallus, is forms the anthrozoid. The anthrozoid and the egg fuse again to form a zygote. Now let us talk about the classification. This classification of pteridophytes is based on the rules and regulations of ICBN. It was given by Smith, and the various divisions are the Xylophyta, which includes the xylophytes, that is Rhynia and Xylotum. The Lycophyta includes the lycopods like Slaginella, Isoetes, and Lycopodium. The Sphenophyta includes the horsetail, the equisetum, and calamites. The Filicophyta includes the ferns like Marsilia, Pteridium, Pteris, Ophioglossum, Azolla, Ediantum. Okay, so then we talk about the stellar evolution. In the stellar evolution, you know that there are two kinds of steel: protosteel and siphonosteel. The protosteel, there are types of like haplosteel, actinosteel, lactosteel. You should be also knowing how the peat developed in siphonosteel. Then two kinds of siphonosteel without the leaf gap and the leaf ridge, that is ectofloic siphonosteel and amphifloic siphonosteel. Amphifloic siphonosteel is also called as solenosteel. Then dictyosteel and eusteel, eusteel in equisetum. We have seen that it was in the internode. Okay, so that is a different thing. Then dictyosteel and then atactosteel, which is seen in monocots. So you should be familiarized with this steel concept and what are the different genres that contains different kinds of steel. You should be thorough with it. Then we move on to the type genus studies. And in the previous section, I have placed about the Rhinia. Rhinia, which is a fossil plant now, and it lacks rhizoid from where the question was asked in the previous exam. And then there are the different fossils that we have discovered, and it was first found in the Rhynie village of Scotland. That's why the name Rhinia came. So this was all about Rhinia. Let us talk about Xylota. In the xylotam, you should know the classification as well as the life cycle. The life cycle starts with the sporophyte, as is the main plant body. The sporophyte, which is diploid, it forms synangium. Then inside which the spore mother cells are formed. These spore mother cells undergo rotational division to produce spores. These spores form the prothallus, which is haploid. This prothallus will form entheridium and archegonium, which are the female and the male sex organs. This will form gametes, and the gametes fuse to form again the zygote. That the zygote will form with divide to form embryo. So this is the life cycle of Xylotum. Let us talk about the next genus, and that is Selaginella. In Selaginella, you should also know about the life cycle, and that is important. The life cycle starts with the mature plant, which is the sporophyte, again diploid. This sporophyte forms the strobilus. This strobilus contains the microsporangium and the megasporangium. The arrangement of microsporangium and the megasporangium is important, and the microsporangium will produce microspores. The megasporangium will produce megaspores. These spores will be produced after reductional divisions, and the microspore will germinate to form microgametophyte, whereas the megaspore will germinate to form megagametophyte. Then the microgametophyte will form entheridium. Megagametophyte will form archegonium. Then the anthrozoids and eggs as gametes are formed. These will fuse to form a zygote. Then the zygote will form young sporophyte. The young sporophyte will give rise to a mature sporophyte. So this is the life cycle that you should be aware with. Then let us talk about the strobilus of this is the strobilus of the equisetum. Then you can of the Selaginella, in which you can see the placement of the microsporangium and the megasporangium that I was talking before. So you should be aware with the how the different species of the Flagellata show different arrangement of the sporangium. So you should be aware with it. You can expect a question from here. Then we talk about the internode of equisetum, which is important. So you should be knowing about the ridges and the furrows, and opposite ridge are present the vascular bundles, then the carinal canals and the valvular canals. The valvular canals are present just below the furrows. So these are the different details that you should should be knowing about. So TS of internode of the equisetum is quite important. Let us talk about the life cycle of equisetum. So equisetum life cycle also starts with the sporophyte, as the main plant body is a sporophyte, which is diploid. It forms strobilus. The strobilus forms the sporangium. The sporangium forms the spore mother cells. Inside the sporangium is the spore mother cells. Spore mother cells undergo reductional division to form spores. These spores give rise to prothallus. The prothallus forms the male sex organ and the female sex organ, which is the antheridium and the archegonium. They produce respective gametes, which is the anthrozoid and the egg. They fertilize, fuse, and then fertilize to form zygote, and the zygote gives rise to a new sporophyte, which is again diploid. Now let us talk about the next type genus, which is the Marsilia. Now in the Marsilia, the cups are there, and various cut section of this fruiting body is quite important. The sporocarps are there, and the how the megasporangias and the microsporangias are arranged, and how they look in different LS and DLS of the sporocarp. So that is important. You can expect a question from there. Then the life cycle part of Marsilia is also important, where the main plant body is a sporophyte, which produces sporocarp, and this sporocarp is contains the microsporangium and the megasporangiums. The microsporangium has got microspore mother cell, whereas the megasporangium contains the megaspore mother cell. The both the microspore mother cell as well as megaspore mother cell undergo reductional division to produce microspores and megaspore. Number of microspores are large. They are mostly 64, and only one of the megaspore will survive. Okay, since four are formed, three degenerate. The microspores give rise to microgametophytes, whereas one of the megaspore that is functional forms the megagametophyte. Megagametophyte produces the egg, whereas microgametophyte produces the anthrozoids. Anthrozoid fuse with the egg to form fuse and then fertilize to form the zygote. Zygote is the first sporophytic generation cell, and that forms the embryo, and the embryo gives rise to sporophyte. Then we talk about this telome theory is important. To just try to read it once again so that you may attend the question. So telome theory is also important, that with the exam point of view. Then we discuss the life cycle of pteridium. In the life cycle of pteridium, a heart-shaped prothallus is formed. Heart-shaped prothallus. Okay, heart-shaped prothallus. So this is formed. So this is quite important, and it you will see in Pteris and Pteridium. Then talking about azolla, then and in the azolla, which is called as water fawn or a free floating plant, in this the stem is floating and it is covered by leaves. So leaves is quite important. You can expect a question from leaves, as the leaves have got an upper lobe and a lower lobe. The upper lobe is chlorophyllous and floating, and the upper lobe has got the cavities in which cyanobacteria and anabenas are present, and it exchanges the it fixes the atmospheric nitrogen for the plant. And the upper lobe and the lower lobes of the leaf are important, and leaves the submerged lobe is called the lower lobe, and which absorbs different which functions as basically as root. So in azolla leaves are important. Then we talk about terrace or pteridium. In terrace or pteridium, one leaf is very compound, is a compound leaf. So each compound leaf will contain several leaflets or the pinna. So a pinna is usually fertile. Now, if you look at a fertile pinna, then it contains Cenosaurus, or the various sories are present, as you can see in the diagram. The sories are present, which represents the sporangias, continuous layer of the sporangia at the lower side of the margin of the leaf. Okay, so you should be knowing about sories, Cenosaurus. What is that? So these parts. Let us talk about gymnosperms. So this is the generalized life cycle of gymnosperms. The It starts with adult sporophyte. The main plant body is adult sporophyte or the sporophyte, which is diploid, and it produces strobilus, the cone, the male cone or the male strobilus, and the female strobilus. These strobilus contains microsporangium and megasporangium respectively. Microsporangiums produce has got have got microspore mother cells, whereas inside the megasporangiums are present the megaspore mother cells. Both microspore mother cells as well as the megaspore mother cells undergo reductional division to produce megaspore as well as the microspores. These megaspore will form female gametophyte, microspores forms the male gametophyte. Female gametophyte produces egg, whereas the male gametophyte produces the sperms. The egg and the sperm fuse with each other and fertilize to form zygote. This zygote will form seed, and then will seed will germinate to give rise to a new adult sporophyte. So this is the basic generalized life cycle of the uh, gymnosperms. Let us talk about the the classification which was given by Spawn in 1965. Now what he did was he divided the gymnosperms into three main classes, that is Cycadopsida, Coniferopsida, and Nitopsida. Now in the Psychedopsida, there are three orders, that is spermales, and it includes the genus Glossopteris, the Bennettitales, then it includes Psychodioidea and the Williamsoni. Then the third order is Pentoxylales and includes Pentoxylum. The second class is the Coniferopsida. It has got three orders. The Chordatates, which includes chordates. The Coniferales includes the Pinus and the Abies. Taxales includes the Taxus. The fourth order is the Ginkgoales, that is the ginkgo. Okay, and here also in the Psychedopsida, here the fourth order is the Cycadales, that includes the Cycas. So these are the classification that you should be knowing. The Nitopsida includes the Nitales, that includes Netum, Ephedra, and Wilwichia. So this is the classification. Just have a quick look at this classification. So it will be easier to identify the genus from the options of the MCQs. Then let us talk about the generalized characters of the gymnosperm. That is, U-stele is present in the gymnosperm. They show secondary growth. They are heterosporous. That is, more than one kind of spores are formed, and gametophytes are highly reduced. They are simple, they are non-green, and they are dependent on the sporophyte. Whereas if we talk about megaspores, they remain permanently inside the megasporangium, and they produce female gametophyte within the megasporangium, and the megasporangium is represented by nucellus. The megasporangium is protected by the integument, and the archegonial neck is very small and it lacks the neck canal cells. Free nuclear division is seen in early embryony. This is quite important. Then it produces seeds which are naked because they are not present inside the fruit. Then there are five species of cycas which are important. But in these five species, three I'm going to discuss that is important for the exam point of view. The first one to discuss is Cycas succinalis. It is found in Western Ghats and Sri Lanka. The young shoots are edible. This Cycas succinalis is commonly called as Crozier cycas, the leaves of which are used in making mats in the southern India. And this plant is also used as an ornament. Now, flour of the seeds is used in making idiom podi, that is famous in South Malabar region of India, and it is used in making cakes and porridges. Young leaves are cooked as vegetables also. The other species that is important is Cycas rumphi, which is also commonly called as Rumphius cycas, and it is found in Andaman and Nicobar. The cooked seeds are eaten well by the hill tribes of Assam. If the uncooked seeds are taken up, they are poisonous. They can cause even death. Now, these Cycas rumphi also yields a kind of gum, which is very important. Then the last species to discuss is the Sagocycus, that is Cycas revoluti. This is the most important species because it yields sago. Now, this is native to China and Japan and found in the and is cultivated in India. Yields sago, which is very rich starch food, and it is mainly cultivated in Japan. It is staple food for many people. Now, sago is extracted from the trunk by removing the bark. Seeds are edible and they contain protein in them. Now, this has got some aesthetic value also because this plant is used in funeral in Europe region. So just go through the economic importance of gymnosperm. Let us now discuss the comparative features of the cycas with the pinus. So a sporophyte of the cycas appears to be a palm-like tree, whereas the pinus appears to be much branched and a very tall tree. Whereas if we talk about roots, then the cycas has got colloid roots, whereas mycorrhizal, ectomycorrhiza, and endomycorrhiza are seen in the pinus. Then if you talk about a stem, the shape of the stem is columnar and branched in the case of cycas, where it is cylindrical and branched in the case of pinus. And it contains shoot, dwarf shoots, and long shoots, whereas persistent leaf bases are seen in the stem. If we talk about leaves, then they are pinnately compound leaves with succinate pinnation in the case of cycas, whereas needle-shaped leaves are seen and they do not show succinate pinnation. Resin ducts are completely absent in cycas, whereas that they are present in the pinus. If you talk about mucilage duct, then mucilage ducts are present in the mesophyll tissues, whereas they are absent in pinus. If you talk about root vascular tissues or the vasculature or the stele of the root of cycas, then they range from diarch to tetrarch and diarch to hexarch in the pinus. If you talk about secondary growth, then primary cambium is very short-lived in the case of cycas, and successive cambial rings are formed from the concentric zones of the xylem, whereas the persistent primary cambium is present, and cambium keeps on producing a continuous cylinder of secondary xylem. Annual rings are absent in the case of cycas, whereas they are present in the case of pinus. Now, mesophyll tissues forms distinct layers as palisade and spongy parenchyma in the case of cycas, whereas such details are absent, like distinct layers like palisade and sponges are absent in the case of pinus. Transfusion tissues are well developed in the case of cycas, whereas they are less developed in the case of pinus. Vegetative reproduction is via bulbils in seen only in cycas, whereas it is absent in pinus. Sexuality of the plant, if we discuss, then dioecious with unisexual cones are present in the cycas, whereas it is monoecious, that is pinus monoecious with unisexual cones. The male cones are very large and terminal in position in the case of cycas, whereas the male cones in the case of pinus are small and grouped in clusters. And if we talk about microsporophylls, then it is triangular woody structure having several microsporangia in the case of cycas, whereas it is a scaly structure and it bears only two microsporangias. Now, microspores are boat-shaped and they are not winged, whereas in the case of pinus, initially it is spherical, then it later becomes winged. So winged, they are winged. That is very important that pollen grains are winged. Now, female cone has got a whorl of loosely arranged microsporophylls, whereas we see an inflorescence in case of pinus. Now, if we talk about ovule, then it is sessile or subsessile in the case of cycas, where it is strictly sessile in the case of pinus. Micropyle in the case of cycas is present on the tip of the sporophyte, whereas it is present on the at the base of the scale in the case of pinus. Micropylar tube is present, whereas it is absent in pinus. Male gametes are motile in the case of cycas, whereas they are non-motile. Pollen tube functions as ostomium, whereas it only functions as sperm carrier in the case of pinus. Female gametophyte may range from may contain 3 to 6 archegonia, whereas 2 archegonias are present in pinus. Pro-embryo, there are no distinct tears that are formed, but 4 tears are formed in the case of pinus. Embryo is only 1 embryo is formed, whereas 4 embryos are formed. Cleavage is absent in the case of cycas, whereas it is present in the case of pinus. Seeds, I have got two cotyledons, perisperms is absent, and it is not winged, whereas the seeds are numerous in the with numerous cotyledons in the case of pinus with perisperm present, and they are winged also because of the pollen grains that are winged. So this was the details, differences between the cycas and the pinus. Let us talk about the order details, which includes ephedra and netum. Now these are the woody plants, which includes the netum and the lianas, the shrubs, as well as uh, stumpy turnip-like plant like Wilvetschia mirabilis. Now, leaves are simple, elliptical, and strap-shaped, and they can be reduced to minute scales. They are mostly generally opposite or whorled. The most important point in the case of nettles is that the vessels present in them as in the case of secondary wood. So secondary wood has got vessels. Now are unisexual and mostly they are usually dioecious and only rarely they are found monoecious plant in the case of netum. Now, flowers are arranged in compound strobili or the inflorescence. That is also an important feature. If you talk about male flowers, then male flowers are surrounded by perianth, and each flower contains an anthrophore and 1 to 8 synangias. Now, one erect orthotropous ovule is present in each female flower. Nucellus in the ovule is surrounded by 2 to 3 envelopes, whereas micropyle of the ovule is projected in the form of long bristle-like tube. At the time of fertilization, pollen tube contains only two male nuclei, whereas one unicellular primary suspensor cell is present inside the embryo. There are two cotyledons present in the embryo. So these are the key characteristics of the order Natales. Just go through them. You should know about the differences between Cycas and Pinus and Ephedra and Netum main characteristics. Now these are the different life cycle, and this life cycle is for the Cycas, whereas this life cycle is for the Pinus. In the Pinus, you can see that in the Pinus, you can see here are the winged pollen grains that are formed, and in Cycas, they there are two trees that grow, and they grow before sexual reproduction. They grow for many years, so there is very late reproduction that takes place. And you should also know that in Cycas, largest ovule is present, largest egg cell is formed. So these are the different details that you should be familiarized with. Let us now see, just quickly see the life cycle of Netum and Ephedra. In the life cycle of Netum and Ephedra, the only two things that you have to remember is that in the Netum, how the male shoot and female shoot are arranged, and what is the position of male flowers, female flowers are absent, and the Ephedra, how the male and the female flowers are present together, whether they are present on a different branch. So the arrangement of the flowers are important in the case of Ephedra and Netum. Just go through them. Hello, everyone. Let us now talk about embryology. Now, in embryology, you already know that there is a sexual cycle that occurs in the case of angiosperms. Now, this sexual cycle involves a reproductive cycle which has a short gametophytic phase, which is haploid. Now, this produces heterospores, or two kinds of spores, that is the microspores and the megaspores. Whereas there is a sporophytic generation, the sporophyte bears spore producing organs, and they produce spores by the process of sporogenesis, that is the microsporogenesis and the megasporogenesis. Now have a look at this figure. In this figure, you can see how the anthers are formed, and the shape of the anther, and it's a tetrasporangiate structure, two-lobed structure. So you should know that. And inside the anther, what are the anther walls? The tapetum is very important. You should know the function of tapetum, what it does, how the microsporogenesis and megasporogenesis takes place. This process you should be knowing, and how the ovule is placed in the carpel, sigma style and ovary, and then the pollen tube enters, and the pollen tube enters through micropyle and brings about successful fertilization. So you should be knowing these things, and then you can have a look at this figure. This will clarify many things, and this can fetch you question. Now let us talk about the main differences, key differences between the megasporogenesis and the microsporogenesis. So microsporogenesis is meiotic formation, and in this haploid microspores are formed from the diploid microspore mother cell, whereas in the megasporogenesis, this is also a meiotic formation in which the haploid megaspores are formed from the diploid megaspore mother cells. Okay, so the formation of the microspores in are in a tetrad, and they are the placement is tetrahedral. Whereas in the case of megasporogenesis, the arrangement of megaspores in a tetrad is what, linear. So the arrangement is different, and that is important. Sometimes the question may be framed through this, the arrangement of the spores inside the tetrad. Now all the four microspores of a tetrad are functional, whereas in megaspore only the one megaspore is functional. The rest of the three degenerate. The microsporogenesis takes place in microsporangium, and the megasporogenesis takes place in the megasporangium. A large number of microspore mother cells are functional in the microsporangium, whereas a single megaspore mother cell is functional in the megasporangium. This is also very important. Okay, so you should know, have the basic idea or the differences between the microsporogenesis and the megasporogenesis, and the place where it takes place. Now in this slide, you can see the development of the embryos embryo sac, and that is important. That leads to the formation of a seven-celled, eight nucleate structure. Eight nuclei होते हैं इसमें mature embryo sac में. So eight nucleate, seven-celled structure, and there are at the chalazion end there are three antipodals. And what are the function of antipodals? Then two polar nuclei has which will later produce fuse to form a diploid nucleus, and that will be present in the central cell. And later on, this will lead to the formation of triple fusion when one of the sperm nucleus fuses with it. and it will form the endosperm. Okay, so this is the formation of triple fusion or the endosperm, which is a triploid product. Now, coming on to the micropylar end, has got a filiform apparatus which guides the pollen tube, and then there are two synergies that are present, and one egg cell that is present. So two synergies, one egg. As soon as the pollen tube enters, the one of the synergids die out, and it provides a seed for the entering of the pollen tube, that you already know. Then pollen tube bursts and discharges its contents, that is the two sperms, and one of the sperms fuses with the egg, one of the sperm fuses with the pollen nuclei which have already fused. Okay, so endosperm nucleus से fuse होता है, and that makes the triple fusion, and this is the syngamy, that is the fusion between the sperm cell and the egg nucleus, and this is syngamy and this is triple fusion, and these two occurring together, this is called as double fertilization. So these are the terms that you should be familiar with, you should know. Then we talk about microsporogenesis and microgametogenesis. Microsporogenesis is the formation of the microspores in a tetrad, which are tetrahedrally placed. Okay, then from out of these, how the sperm nucleus are formed, then vegetative generative cell, how these are formed, that is important, and that is called as microgametogenesis. Okay, so this is the thing that you should be knowing, and the difference between the two. Then this is a diagram that shows you the complete development, that is the microsporogenesis, microgametogenesis. Here, megasporogenesis, that is the production of megaspore, and then megagametogenesis. So this is the megasporogenesis, and this is megagametogenesis. Later on, this is showing you the placement of the embryo sac as well as the entry of the pollen tube. So this also shows you the fertilization and the fusion that takes place. And just have a look at this complete picture. This gives you a very good idea about how the pollination and reproduction takes place in the angiosperms. So let us now talk about the pollination. So pollination can be of two types: self pollination, and the second one is the cross pollination, or the allogamy, or the xenogamy. Okay, then self pollination can be of two types: autogamy and geitonogamy. That you should be knowing the difference. Have a look at the slide. I have already put in the various details over here. Then the abiotic agencies that bring about, that is wind pollination is known as anemophily, and water pollination is called as hydrophily. This the cross pollination can be brought about by biotic agencies that may involve insect, which is called entomophily. Ornithophily is involving is involving birds. Chiroptophily is by through bats, and the malacophily is through mollusk. Okay, so these are the things that you should be knowing, and these are important for your exam. You can it can fetch you question over here in the part of embryology. Let us now talk about self incompatibility, and self incompatibility is very important. These are of two type: gametophytic and sporophytic self incompatibility, and you should be knowing the mechanism, how the alleles that are present at a locus are involved in are involved in self incompatibility, so that the plant is promoted for the cross pollination. So this figure gives out you the self incompatibility details, and just go through this, and this figure is quite explanatory in itself. You can just see that the how and when the pollen grain or will be able to form the pollen tube, and the pollen tube will be successfully able to grow inside the style, and will enter the ovary. Okay, so these are the details that I wanted to share regarding embryology. So just go through this, and you should also be knowing the mechanism of self incompatibility. That is also important. Just go through this mechanism of self incompatibility. Okay, this was the last thing that I wanted you to have a look at it. Then just prepare well and do well in your exam. Good luck, students.